So audio test 1, 2, 3.
All right, you're good now.
Perfect. Thank you very much, Tara. All right, so let's go ahead and do some introductions.
All right. Good morning, everyone, and aloha. This is your friendly biology lead, Megan Putz, and I'm joined here today by my colleague. We're both from the University of Hawaii, and we're excited to be here with you on the Okeanos Explorer. We're exploring Beethoven Ridge today. Dun, 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 dun. Dun, 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 dun. And we'll be investigating some of the similarities and differences in the community composition of our deep-sea corals and sponge communities on these ridge features within the musician seamounts. It's going to be really interesting to see how these communities on ridges compare to other ones in different locations along the seamount chain and compared to different seamounts within the chain.
So who do we have joining us today on the line?
Do that. Oh, well.
That was a really good geology that I heard.
Tara Harmer-Luke from Stockton University in New Jersey.
Welcome, Tara.
Hi, it's Chris Kelly calling in from home, at least for a little while today.
Good morning, Chris.
Morning, Chris. Morning, Tara.
Morning.
So is anybody else joining us today? All right, well, John, will you give us a little bit of background about the geology that we're going to likely see today, please?
Sure. So yesterday, if you were with us, we dove on a small volcanic ridge, small relative to what we're diving on today, to the south of here. And these ridges are referred to as volcanic elongated ridges in the literature. They're kind of unusual. They're thought to have formed from interaction of the hotspot plume, the hotspot trace that created the single point musician seamounts, the kind of a traditional conical individual isolated seamounts in a chain, versus our interaction with this mid-ocean ridge system that created these constructional volcanic features that basically erupted from the seafloor. Unlike the larger tectonically derived fracture zones to the north of here. So these are very long ridges. They're very relatively steep, and then on top of them there's a number of volcanic cones, kind of like mini seamounts, that will be actually is one that we'll be diving on today, a small cone at the summit of this ridge. So
some fun facts about Beethoven. He was born in Germany in December in 1770, but no one really knows what his exact birthday date was, so we just have an estimate of when he was born.
So we do have bottom in sight.
Oh, wonderful. Even better. Another fun fact about Beethoven that I learned yesterday when I was reading our dive plan email, that Beethoven actually used to dunk his head in cold water before composing his music. Perhaps he's trying to channel some of that deep sea mystery into his music.
I was just going to say, I do the same thing before all of our dives.
Right?
Wonder if Beethoven used salt water, though.
You got to get in the mood somehow.
All right, before we get going, I saw that also Asako Matsumoto joined us in the chat room. She's not on the line, but she's in Japan, so Ohayo gozaimasu, Sako. So everyone, just be patient with us while we get the ROV stabilized and get ready to start doing some science.
Hooray, science! I'm so excited. Well, I don't quite see any biology yet. Hopefully soon. Sometimes these organisms can be very small or hard to spot from far away, so we're going to have to wait a little bit till we get situated on the bottom and start exploring.
Roger that.
Go ahead, Arvina. We have the vehicles on the bottom. Okay, thank you.
Alright, then you can go ahead and put EXRV back on, John.
Copy that.
You can zoom past the skids rolling. Skids rolling, yep. Thanks.
Coming down with you.
Yep. It's a little barren.
So this looks a lot like the bottom we saw yesterday. A lot of sort of pebbly, cobbly, interspersed with sediment.
The depth and high pack.
Yeah, this one's a little
CTD
less
nice.
uniform. Yesterday we landed and it looked
USBL is
like it
usually
was
10 meters off or
kind
something.
of a consistent sized rubble and
Where do we get the
similar shape. These are
depth?
The parasite.
variety of
23.
shapes,
Okay.
sediment
What are
and
you
light
reading
sediment
there?
in between.
2534.
Plus 3.
Perhaps we're seeing this because we're at the bottom of that cone, and as we start transecting up that cone, we'll be able to see a little bit different types of geology.
Yeah, from yesterday there was a dramatic change in the geological contact between the two types of substrate or bottom.
So actually we're noticing a bit of a current coming from starboard to port, which would be roughly south to north. In case that's helpful for you.
Okay, thank you. So we're noticing a bit of a current right
Okay.
now
Push
from
it out a
south
little
to
bit.
north.
We'll probably want to get a move in here pretty soon.
I have general uphill sort of at 100.
100. Okay. You can turn toward 100 then.
Copy that. Nav, let us know when you're done with your
So do you think this area would be a
Bottom
good
numbers,
place
we can
for
get a move going.
rock collection, or should we wait until we get further up the slope?
Well, the rocks actually look pretty good.
Pull your swing arm.
Yeah, I like this one right in the
Sounds
middle. I don't know.
Um,
It just looks nice to me. It might be too big.
Yeah, a bit of a current here.
It does look pretty hefty.
Sort of like the one we got yesterday.
Yeah, there's actually a few. There's one to the upper left that might be good.
Upper left?
Yeah.
Are the
The
lasers
one
on?
in the center screen or up
I
above
don't
it?
think so.
What's that?
Pilot, are the lasers on? Oh, I
The
see them.
lasers are
Thank
on
you.
now, yep.
Yes, the lasers are on now, Chris.
Is it the one to the right of the lasers or upper left still?
Upper left.
Okay.
I'm not sure if that's one or two rocks or three.
Just moving ahead a little bit here, Jeff.
Copy that.
Yeah, one of those, but they may be a little big. One of those two would be good if they're not too large.
Okay, let's go full wide, Roland. Thank you. So the ones that the laser is on now, or just to the right of it? There? That looks loose, John. So one of these two, they both look loose.
Yeah, how about to the right there? That one
right?
kind of sticking up, looks like it's on edge. Yeah, that one there.
Above this one here, or above it?
Either one of those two.
Either one of them.
Yeah.
They're both loose, so just let me know which one you want. We can get either. We can do a quick zoom once I set down. You can choose from there.
It's really important to grab a rock right away. That way, if we don't see one, we don't have to worry about it.
Yeah, we kind of waited a little long yesterday, and then we got into an area that they were all cemented in, or volcanic, or intact volcanic flanks. So.
All right, pilot.
Sampling of opportunity.
Okay,
Would you like me to take mini?
Sure.
Put it
Let's
up
do a
on
quick
the screen.
zoom, Roland, and then that's good. So upper or lower, John?
Well, I'll go for the one on the lower one if it's if it'll fit.
Yeah, I think that's reasonable.
All right, your mini Zeus has the arm and the rock. Let me know when you want to adjust it.
Okay. Should be fine. I guess I'll just go ahead and go with the starboard wing.
Starboard box.
Yep.
Okay. Yeah,
So-
it looks clear.
Okay.
You want me to move it in after you sample?
Yeah.
Okay.
Force feedback is on. Pots look stable. Indexed out. Alright, hydraulics coming on. Okay, let's come out a bit, Roland. I do want to take my Zeus Plus and tilt up a bit. Okay, John, you want to zoom in, Roland? That's good, right there. What do you think?
Oh, well, it's got a little sponge on it.
Right on the bottom there?
On the bottom there. Let's take a nice look at that before we put it in the box.
Go ahead, Roland.
Chris, do you know what kind of sponge this might be? It's got like these little hair-like.
Would you mind if I adjust your starboard
Oh, and there's
upper?
something else on it. It's
The
really,
swing arm?
Yeah.
Yeah.
really
light up
interesting
your box.
spicules. So I think this is a glass sponge. Maybe.
I'm not seeing the sponge right now, but I'm delayed a bit.
Yeah, just give it a few seconds. It should be coming into view shortly. It looks like a little tube, and it's got these sort of hook-like spicules sticking out all over.
Okay. Do you want to zoom out, Megan, or you want to
Yeah.
That's good. Thank
Okay.
you,
Yeah,
pilot.
let's,
go ahead and get it in the box?
Yes, please.
Okay.
yeah, looks good.
Won't do any more rotation.
Sounds like they're
Alright.
happy with that. I can do a quick one here, Roland.
See if there's any other commensals on it.
Sure. Oh, there's something on the... What's that?
Goo.
Nope. Little.
Looks like a creature, doesn't it?
Well,
Ah.
I'm spoiled with that video
Oh, yeah.
wall at the UHE.
That is
I
a
just
strange.
didn't see that from my home laptop.
There's some sort of gelatinous
It
creature.
tilts up a touch. Sure.
I have no idea what that is. That is really interesting.
There we go.
Some
It
kind
looks
of
like it has
ctenophore?
tentacles. I don't think it's a ctenophore. Ctenophores generally have some sort of like 2 openings, and this opening has like a ring of tentacles around it. Maybe it might be some sort of anemone, but it looks really strange.
Looks like a cube with a round hole in it
Yeah.
from this angle.
It almost looks like something like pelagic that would be floating around in the water column, but it's stuck onto this rock.
Unfortunately,
All right.
I'm not sure it's going to make it up to the
Okay,
surface.
I'm seeing the sponge now.
So if you want to get some more good views on it, we can
I thought
rotate
I
it a
was
bit.
on the 7-second feed, but that took a while to
Let's see
show
if we
up.
can look straight down on the top of
Anyway,
it.
it looks like, well, it's a sponge,
Want me
I
to
think,
zoom down a
but I've
little?
never seen anything like it.
Got it.
We can always hope it'll come up intact with this rock.
That's good.
Of this little gelatinous
It's different.
creature that we're looking at right now. It almost looks completely empty, and I see a little amphipod, perhaps,
Yeah.
on top of it, maybe living inside of it.
Okay, Roland. Let's come out.
All right.
Ready for
Pilot,
starboard
is this
wing?
going
Sure.
starboard, forward?
Starboard forward.
Roger.
All right, starboard wing inboard.
Is it too big for a bio box?
Yeah,
Yeah.
that's good. Carl, thank
Okay.
you.
All right, I'm seeing the gelatinous creature now,
no Andy
and
rock,
I have no clue.
good. I think
either.
it's perfect.
First
It's
time
probably
I've seen
25
anything
pounds
like
or
that. That's
so.
pretty wild.
I know, isn't that weird? I just can't think of what it is, but you know, you saw that ring of
How do you like your lighting
tentacles
there?
around the
I brought that
opening.
upper in.
Good.
Looks much
So it
better.
reminded me of an enemy, but everything else about it is, you know, shouting, you know, some sort of other type of Nidarian.
So it's hard to even figure out what's up and down on this thing. I mean, it could be some type of a weird hydrozoa, and I think they're the type
Looks
that
secure.
kind of settle down on the rocks,
Bring,
if I remember
Vinny's
from the
just
last
back
guide
over
that somebody was
Roland likes
talking
it.
about. I can't even say for sure
Got
that.
that back.
But
Yep.
yeah,
Yeah,
again,
I'll give you
let's
both
just
your
hope,
cameras.
some of it
And
comes
right
up.
screen.
It would be really interesting to get a
Serious.
assessment of that.
Alright.
Yeah, definitely. Well, we'll see. Maybe it'll make it to the surface, but probably not. It looks way too fragile for that.
Thanks, Carl.
Thank you.
A little bit to the left of the middle, top of the screen.
See the whip?
Oh, I think I see it. Yeah.
Yeah, there's a single stalk of
Yeah.
what I think might be a primnoid coral. We're just gonna take a zoom on to that soon, see if it is what I think it is.
It's a little bit out there. Let's go ahead and we can get 20 meters in. So, Jeff, we're going to need a ship move to get up to the coral that they're talking about. I don't think I'm going to be able to get out there. I'll try. But 105 looks good to you, or 100?
Either one, yep. 105 is fine.
Okay.
And I come down and give you a little extra.
Yeah,
room.
that'd be good.
Jeff, can you reposition HD2 when you get a chance?
I will.
Isn't that where you like it, Roland? Between the toes?
Right now it's still angled, sort of sideways.
He's pilot's
Okay.
got it.
How's that?
It's getting there. Little,
You want it right in the center?
Yeah. There we go.
How's that?
It's good.
Okay. Okay.
Lets us to see things coming, or
Okay, altitude 15. I'm going to call that about as low as I want to go.
Yeah, that's fine. I think I can get to it here.
Yeah, we should get that move going
Yeah.
anyways.
Yeah. Once he's done.
He's still chasing numbers.
Okay, you want to go in for a zoom?
Now, co-pilot,
Go ahead, Copilot.
how's it going over there?
All right, we're
Good,
zooming
just
in
making sure we got all your
on
readings for you.
this
We were missing the,
from NOAA
CTV
coral.
temperature for the sled up on our
I
display
think it is a Norella.
I think we're
So the
getting ready
Norella
for a ship
Primnoas
move. You ready
have
to help us with that?
Excellent,
these
absolutely.
large
I think we could do at least
3-tiered
30
body
meters.
Yeah,
scales.
let's do 30.
At 105.
These
At
ones seem
105?
relatively large.
Yep.
Chris, do you think this might be Norella
And at 0.2 knots,
macrocalyx?
okay.
210 sounds good, yep.
Sounds good, I'll call that in.
Thanks.
Okay, is that good for an ID, Megan?
Yes, pilot, that's
Okay,
good.
let's
let's
zoom
go
out.
full screen.
Bridge ROB nav.
Go ahead,
I
RV
just took off, Roland.
We're
but
ready
I'll
for our
set
first
back
ship
down
move
for you.
if you
That's okay, you're stable enough there.
Excellent.
Lasers on.
We would like to go 30 meters, 30
And
meters.
getting lasers,
Bearing
We got
105
lasers earlier.
full
Okay.
line.
at
Thank you.
0.2 knots.
30 meters.
Just gonna
bearing
back up a little bit,
105,
Jeff.
3.2.
So if this is Norella macrocalyx,
You got
it's
it.
probably relatively
Thank you.
young. It hasn't branched yet. But it might be another type of unbranched
past the
Primnoa
schedule.
coral. We collected one yesterday at the beginning of our dive that was of possibly a new genus that has not been identified yet.
Let's just take a peek side to side here. See what the slope looks
RV
like.
nav bridge move of 30 meters bearing 105.
Tilt up a little higher.
Thank you, Bridge.
What?
Yeah, I don't really want to do that because then science will want to go to something we can't see.
I haven't seen
Or
anything
can't
yet.
get to.
Should move on
I'm
bridge.
even looking. Yeah.
But we're holding our fingers, crossing them for some octopuses today. We didn't see octopuses yesterday for Octopus Friday, but you know, maybe it's a delayed reaction.
It's a rather large boulder rock there.
Well, now that we've got a rock, I think it might be prudent to start heading up slope.
Yep, we've got a ship move in.
Awesome.
And we're just taking a peek at what the slope looks like, and then we'll make our way up.
Perfect.
Yeah, so Megan, I would concur it's Norella. I'm not sure about macrocalyx. There is an unbranched Norella that's in Hawaiian waters. It's Norella hawaiiensis,
Going and back up.
interestingly
That's
enough.
pretty,
But I just can't
benign
remember how many polyps per whirl it was and other features of it. But I would definitely agree with your genus. Looks like Norella simulates.
Go ahead now.
Thanks, Chris.
If you don't mind,
Yeah,
let me
there's
know when you
there's a lot of
think
different
you're
species
moving, and
out
we'll try and
there,
gauge how long it
but
takes
it's just
at this
for
step.
some reason the macrocalyx
Yeah,
always
so
sticks
I
in my
think-
mind with having those sort of
There's a
almost
shrimp.
cup-like body scales. So it reminded me of it, but I'll have to refer to my animal guide to see if
you want to zoom in a
it
little?
might be that.
Hold there. They're actually zooming past
All right,
the lasers.
Megan, here's the shrimp. Tell us all about your, I think this is one of your favorites.
Yeah, this is one of my favorites. This is a Pneumatic Sinus shrimp. They tend to always be on the rocks, walking along the bottom with their very long legs.
Do they also have long antennae?
They do have long antennae. So that's what you see coming off the head and going up into the water. He's turned around for us, giving us a good view from multiple sides. So we see these very often at these depths, just chilling out, walking around, doing their thing. Now they're very hard to identify as species because we need to get a good look at how many little spines are on its rostrum, and that's the little projection between the eyes. Kind of looks like a nose. And even with our amazing video, it's really hard to count the individual spines. So having a specimen is one of the is almost required to be able to identify it with a lot of certainty.
Yeah, sorry about that. Roll in. Let's come out. Side and down slope. So what makes those your favorite shrimp, Megan?
I just really like shrimps. Not only are they delicious, but they're just so cool the way they walk around on the bottom, and then when they start swimming with their little pleopods, they're just cool.
What do you see, Roland?
It just went under the vehicle.
You saw something white?
White. Sort of possibly one of those little worms. It's moving pretty quick. I think it's out of view.
So for those of you who are wondering what we're looking at geologically, this is kind of a typical, what we call a Talus Slope, or some people call it scree. It's this broken rock.
It came and went.
Broken rock these look like they're of volcanic origin from lava flows and pillow flows that are upslope. So these can fall off just from breaking up over time. They can, you could have earthquakes, landslides, so on, and there are a variety of sizes. As you can see, sometimes they're more uniform, sometimes they're all one size larger. You see this on land too, at the base of mountains and cliffs, rock quarries. It's a jumbled collection of rocks, basically.
I see something in the manipulator camera. That may be a fish.
Oh, a fish? It's exciting.
Oh,
I think it might be an eel.
A fish and a...
Oh, and it might be a sponge. So let's get the eel first.
Before I scare it away.
You never know when they want to swim away.
Copy, thank you.
Oh, Bruce was telling us about this guy yesterday. We saw him during the dive.
That's fine.
Do another 30 meters.
So I believe that this eel is an Ileophus, or a sawtooth eel?
Go ahead, RV nav.
What did he say
We'd
it
like to
was?
duplicate our last move if we could. 30 meters, bearing 105.
An Aerotooth eel. So an Aerotooth eel in the family Synaptobranchidae, in the subfamily Ileophini. So this is an Ileophus Aerotooth eel.
What's with all the kind of modeling the lighter colors? Is that battle damage, or is that part of his camouflage? Or
it is damage. A lot of times these eels will try to grab something in between the rocks. So maybe this guy's been, digging around in these rocks looking for something to eat, and he's scraped himself up.
Zoom in tighter.
Ship move underway, pilots.
Bruce might know a little bit more about this, but since it is Saturday, the IRC EEC is not currently open.
what do these guys eat? Do we know?
I'm I'm not sure what they eat, but if I were to make a guess, I would say they probably feed on small invertebrates.
Like shrimp?
Like shrimp or polychaetes.
Those are worms, right?
Those are the worms. So we saw one yesterday that did a little bit of a swim for us, not too much. Perhaps small fishes, but I'm not quite sure.
What time did we get to bottom, John?
We got
bottom
here
at
9:40. So we're coming into what looks like a sponge, Chris.
The left-hand side of the sponge has a...
And it might have
sort of
a
flap.
shrimp on it. Now I think it might be a pheronematid sponge.
Okay, we're going to go partial rolling.
Not shrimps.
Hold there.
Oh wow, there's a lot of little things on here. We have a benthic ctenophore. Looks like it might be what we've been calling Tijfialia or Tijfialia. It's one of those really weird names that has a T and a J right next to it. And ctenophores are generally seen in the water column, but we do have some benthic ones that attach to different substrates.
Sure.
Yeah, so this one does not have little, so the little projections you see coming out are how it captures its food. Those are its long feeding sort of tentacles.
Can they retract those, or?
I believe they can. Their mouth is actually on the underside of the animal that's up against the sponge right now. And so they bring in, sort of like reeling in like a fishing line. And those tentacles have a little bit of stickiness to them to capture something.
Go ahead.
Ah.
And then we have a hydrozoan in the family Tubularidae, and that's this
Actually, do you want to zoom in on that shell up there?
Yep.
little guy on the stalk. And oh, that's another benthic ctenophore. It might be being fed upon by some sort of gastropod or snail.
Wow, we got quite a party going on.
Yeah, it's a party on the sponge.
Okay,
So I think this sponge, it might be Simperella.
And then I'll push ahead.
Can we look at the base of the sponge to see what kind of attachment it has?
Zoom on the base.
So this sponge is in the family Pheronematidae, and I'm not. It's sort of wedged itself in between these rocks, so I can't see the specialized spicules that are allowing it to attach down into the substrate. But it does look to me like a Simperella, and hopefully Chris Kelly will be back shortly to give us a little more information.
So a small coral,
maybe confirm my
right.
identification of this sponge.
Keep an eye on the hard
Roland
returns,
sees something on the right here. See what he's
Oh
seeing.
yeah, let's look at what he's
Also,
seeing.
is that a good range for you on the sonar?
We can bring Sirius into 50.
Copy that.
I'm slipping, rolling. Come out a little bit.
Looking at you, 80 degrees.
All right. We just have a second here. I got to push out. Go ahead.
So this is a very young bamboo coral. And it has extremely long intertentacular spines. So those are those long projections in between the tentacles that are coming out of the polyp. So this one only has 2 polyps, it looks like. So it's a very young coral.
What do they use those spines for?
I don't know if they use them for anything. If Scott was on the line, he might be able to tell us, but I'm not really sure. But those spines are an identifying factor to classify that particular coral into a specific keratoisidinae clade.
You know what I'm thinking. See something else, maybe another sponge up ahead. Nope.
This looks like a black coral in the genus Bathopathes.
Pilot, this is Nav.
This one looks like a relatively large one. If we could zoom in on the stalk of this Bathopathes.
Sure.
Sometimes these corals have an associate polychaete worm living along the stalk.
Can you bring up? Well, is the lighting better with the lower swing arms down like that?
Right now.
And where do Bathopathes live?
What do you think?
Up higher.
On the bathymetry.
All right. Now you're learning.
I'm learning.
I'm going to move around to the side of it here. So we get a better view.
Obviously, the lighting was better low, and we only put them up. It's going to be less steep.
Okay.
But I'll put them up. Right now.
Just for now.
Oh, there's a little crinoid on the rock.
Zoom in.
Yeah, crinoid.
It might be a telecrinidae.
So how do you tell the difference? Are they a number of legs from a distance? Is it just the number of appendages?
So from a distance, it's hard to tell, but the telecrinids tend to have less dense projections on their arms, like they're not as feathery looking. I'm not seeing a polychaete on this stem. Can we pan down to the base?
Tilting down to the base.
And
there
then
it is. There's the crinoid.
let's look at this crinoid. So, yeah, see how spaced out the little feather-like projections are on the arms? And I'm not really seeing the cirri, which are the little legs that are on the underside of the crinoid that help hold it down to the substrate. But those will be relatively long and prehensile.
Okay.
Thank you,
zoom
pilot.
out. Do you want to look at the top of the black coral here?
I think we're good on this black coral.
All right, Roland, are you happy?
One full screen of both specimens.
Okay.
It's a pretty large specimen. So the lasers that you see in view are about 10 centimeters, or exactly 10 centimeters apart.
Thanks, Jeff.
And that's approximately 4 inches.
Okay.
So this
Happy with that,
coral
Roland?
is
Video's
probably,
clear.
All right,
you know,
zoom out.
1.5 feet. What did she say?
Yeah, that sounds about right. From stem to stern.
Yep. Stem to tip.
Quite a bit of current.
Is it picking up as you come uphill?
I don't know if it's getting worse, but it's definitely strong.
So now that we're on our way, it might be a good time to talk a little bit about our mission. So this is our last expedition within the Capstone Project, which is the campaign to address Pacific monument in scientific technology and ocean needs. And it's a really great opportunity to explore all of our Pacific monuments. So we've been here exploring in the Pacific for, was it 3 or 4 years?
3 years.
3 years. And we do this with our partners. A lot of partners and a lot of people from around the world have helped, add to the expedition and make it as amazing as it has been. And those partners include OER.
Seen a whole lot.
If I have to remember, the Global Foundation for Ocean Exploration. NOAA's National Marine Fisheries, Marine National Monuments Program, the Deep Sea Coral Research and Technology Program, and we can't forget all of our scientists from around the world that have called in and helped us with identifications, collections, dive planning. It's really been a great program, and I'm sure I'm forgetting a bunch of different organizations that have added to
What's
our
pace that we need to
great
make today?
capstone program. So I think it's a great time to introduce our front row. Our front row consists of our pilots and navigators, and we also have our videographers. So if you guys wouldn't mind introducing yourselves.
Sure. This is Dan Rogers in the pilot seat, and
sitting co-pilot is Jeffrey Lanning.
Sitting navigator is John Mefford.
And in the far corner, in the shadows, is Roland Bryan.
And in the back clipping is Caitlin Bailey.
And we're all with the Global Foundation for Ocean Exploration. Yeah, I probably want to get a move.
All right, thanks.
Yeah.
Thanks, everyone.
We should be pretty good on pace.
And we
right
also
now.
have a
We've got
whole
about 5 minutes
ship's
total,
crew
but
that's
we've got plenty of bottom time right now.
piloting, maintaining, cooking, cleaning, painting,
Would you like to do the
launching
same
and
move?
recovering. So we have an entire ship's
Yeah.
crew
More the
that
same is perfect.
keeps us going.
Sounds good. I'll get that called in. Bridge ROV nav.
Go ahead.
We'd like to duplicate our previous moves.
Okay, 30 meters, 105, speed decimal 2.
Affirmative.
Okay.
Thank you.
And
So
we
John,
also
just for
have
future reference,
our
it's good
illustrious
you repeated it, but generally we'll
expedition
just say,
coordinator,
even if it's the
Casey,
same
and
move,
we
we'll just
also
repeat it, so
have
there's
a number of data
I was going to
people,
until she
our data
immediately
sample
chimed in.
data manager Nolan, and
No, I
a
was
couple of the other GFOE data managers that keep track of all the stuff that we're collecting.
And we have our mapping lead, Mike, who has been great at, keeping the mood light and finding us great spots to dive on.
Decimal 2 has been initiated.
Copy that.
Yeah.
something. by
Yes,
the
and
way.
his companions, Amanda and Charlie, tech. So they work a lot during the nighttime and when we're not diving. So when we're transiting
That looks like a
also,
really short
and then
one,
when
and
we
this has
arrive
those
on site,
long
producing
spinnacles as well.
the maps that we can work with right away in the morning.
I'm not even putting any lateral on right now.
So we're seeing another coral. Could be either a bamboo coral or a primnoid coral. Let's get a nice snap zoom on this, see what it is.
Do a snap zoom. Roland, go ahead.
So this is another one of those bamboo corals with the very long intertentacular spine. So this one's a little bit older. It has a lot more polyps. Scott, I see that you're in the chat room. Maybe you know what clade this belongs to?
Okay, let's zoom out. You do your full specimen rolling.
So your light pool, or do you want me to turn around? Get you?
Yeah, you can turn a bit. Okay. Thanks, Roland. I'm just going to see what these other whiffs were to the left real quick, then I'll
Yeah, it
move
looked
back
like
in
there
line
was a.
if I move.
Looks like there was a branched one there. That little group that you're headed towards. Or it was two singles close
there's
together.
a sea star.
Alright, I'm about 30 degrees to port.
Okay.
All right.
Your mic is off.
We'll do a quick one. Quick zoom, and then I got to line back
Okay,
up.
so I see that Scott
You're
France
okay.
is saying that the bamboo coral that we just zoomed in on might be part of the bee clade.
Why don't you come out a bit, Roland, and let me sit down here. All right, go ahead.
Interesting substrate.
Oh, this is one of Chris Maw's favorites. It's a Goni Astrid sea star, or cookie cutter star, or cookie star. So they look a lot like little cutouts if you were making cookies from cookie dough. Now I find these really hard to identify. I always have a hard time keeping straight how many side scales and the different types of goniastrids. So maybe if Chris is listening, he might call in and tell us a little more about this star.
Okay. You want to zoom out and roll?
Run the lasers over it real quick. Okay, clear.
Megan,
there he
this
is.
is Chris Ma.
Hi, Chris. It's great to hear from you.
How are you?
I'm doing great.
Well, I chimed in the other day on some coral predators, and was happy to speak to John. This animal that you're looking at here is one of these really neat animals that if you get a few moments towards the end of a dive and you haven't collected anything yet, I would love to see one of those. So this is a Goni Astrid that I'm not entirely sure has a name yet. It might be something called Sibogaster, named after the Siboga research vessel, which explored the deep sea many decades ago. But I'm not sure because I need to look at some of the characters up close. But at this depth, around 2,000 to 3,000 meters, there aren't very many goniastrids, and I've spent a fair amount of time trying to figure out which ones we end up seeing, and this isn't one of them that we see that is really well known from the literature. So that is really, even though it wasn't perhaps as immediately as dynamic as some of the coral predators, it was still very interesting, and like I said, really neat. Ooh, you have a stock crinoid up.
Yeah. Sweet.
So this is, I'm not entirely sure which one this is, which genus this is. It's vaguely Bathycrinus-like, but I'm not sure. I probably am slaughtering that identification terribly. But the one thing to note that is really unusual is the fact that the stalk, you'll notice that the stalk is covered with that sort of fuzzy thing. Those are hydroids. Those are animals. Those are sort of like reminiscent of the hydras that you would find if you were taking a cup of pond water and looking at all the little animals that live in it. They're basically sort of little tiny extended sort of like sea anemones almost, but they're taking advantage of that surface and probably filtering the water as it's going by. It's unusual for crinoids.
I've never seen that before.
Excuse me. It's not unusual now that we've seen crinoids with these animals living on the stalk, but there's technically, I think there's actually supposed to be skin covering that surface. So usually they don't have animals growing on them. So whenever I see this, I always think it's a little odd, but we've seen it now, and I think at least two 2 or 3 different species of stalked crinoids. And this one, I think this is the one that we always see with stalked or hydroids growing on. That's
a little
at least
while ago, yeah.
So, Chris, those are.
it is definite. These are, by the way. I'm sorry.
Oh, are those are individual hydroids, or is it a community or one organism? They're individuals. Is that correct?
Yeah, I believe so. They're not like the octocorals you see. I think they're actually a host of individuals living on the surface. I might be wrong there. This is also just to sort of give a general introduction. The stalked crinoid itself is the kind of older form of those feather stars that we see. Stalked crinoids are sort of very ancient animals. As a geologist, I'm sure you're probably familiar with them because they always show them in paleontology textbooks. But there are fossils that are just like those stalked forms from the Paleozoic. And so they are often reminiscent of very old animals.
Right. Chris, I was seeing some comment from Tina Molodtsova from, I believe she's at the Shirshov Institute in Russia, that it actually says that they are kind of common to have on the stalk, and that this would actually be a colony of hydrozoans rather than individuals, I believe. So.
Okay. Well, I'm happy to acknowledge that I've seen a fair number of hydroids. Common is a relatively difficult word to define. But colonial, sure, that makes sense too. But I leave it to the people that know the stalked crinoids a little better. Anyway, I will leave you guys to it, and if, like I said, if an opportunity to grab another Goni Astrid on one of those rocks comes up, I hope a 2-for-1 presents itself.
Okay.
Yeah, before you go, Chris, I wanted to ask you a question, or maybe you have an idea about this. Mike was just showing me on the EK60 that there is a scattering layer. Now, this image is going to be on video feed 3. There's a scattering layer at about 150 meters off the bottom, and I can't think of why we would see that. Do you have any ideas?
No, I'm not much of an oceanographer, I'm afraid. I could tell you a lot about phylogenetics and systematics and taxonomy and such, but I'm afraid I've never really been really proficient at a lot of the sort of more physical oceanographic things.
Well, thanks. Maybe somebody else on the line or someone will call in and have a little more idea about this. But for everybody in the chat room, maybe you could chime in, take a look at video 3.3.
Okay, zoom out.
So this is Mike White, your friendly mapping lead for folks following along on shore. We're just sitting here. One of our new paradigms is continually running our EK60 split beam, single beam sonars. And what we're just seeing here in the EK18 or 18 kilohertz frequency, I'm going to send a screenshot, is what you can see clearly is this is the ROV as it descends towards the bottom. You want to think of the horizontal axis as time and the vertical axis as depth. So this would be the surface of the water. Here we have the scattering layer beneath the surface, you know, and at night this will migrate up, of course. These vertical lines here are the USBL, the ultra-short baseline, which the ROV team uses to track the ROV in the water. And what we've been noticing here is about 100, give or take 150 to 200 meters above the bottom is a relatively consistent scattering layer that was there before the dive and seems to be there throughout the dive. So if anybody on the line wants to make a comment about this, please. Otherwise, what I'm going to be doing is sending some screenshots to our water column experts that we have here at OER.
So we have a couple of theories coming in on the chat line. Scott France is saying that it might be fish aggregating above the seamount, and Tina Molodtsova is suggesting that it's plankton hiding question mark.
Hold there.
So we're zooming in right now on a sponge. Now this looks like a Bolosoma sponge. It is a euplectellid, I believe.
Looks like a mushroom.
Yeah, it kind of looks like a mushroom. So if you were tuning in yesterday and at the end of the dive, we saw a sponge that had a long stalk that looked like a mushroom as well. This is a different family. This is in the family Euplectellidae in the subfamily Bolosomini.
Okay,
And it's probably the genus Bolosoma. And they're stalked as well, but the base is coming up onto the atrial surface instead of the dermal surface, and those are the different surfaces of the sponge. The atrial surface is the inside, and the dermal surface is the outside surface. And those are just sponge terms to figure out where the stalks are connecting and what is the inside and outside. So here we're seeing another primnoid. And it has a little associate. It looks like possibly another hydrozoan of those tubularity.
A brittle star there.
All right, and while we've had all this biology going on, we've moved into more of an intact flank instead of all this just talus, broken rock. We have more kind of intact lava flows and some sediment in between. And I think the slope's a little steeper here than we were on originally. So we're moving on up, and most of this, or all of this, is really all coated by this manganese crust or polymetallic crust that precipitates out of the seawater. Forms a coating, like a chocolate coating on top of the all the exposed surfaces of rocks, and this grows collects very slowly at like millimeters per millions of years.
A lot of sorts, isn't it?
You know how to get to my geology heart right there with the chocolate.
This is dark chocolate.
Dark
too.
chocolate's
Yeah,
my favorite.
Dark chocolate coating.
Same deal here with the.
So I think this is the same type of stalk crinoid we just looked at.
With the same hydrozoans?
With the same hydrozoans along the stalk. So it is likely in the family Bathycrinidae. Did we have a genus ID? I was talking to Mike when we were looking at it earlier.
No, I don't think we did at the time. I was, let me just look back in the for the crinoid itself, or for the...
yeah, for the crinoid.
No, I don't think so. I think Tina was mentioning about the hydroids, hydroactinia.
Stalks up here. Okay, let's do a snap zoom. Pushing ahead.
Something.
So there's a possibility that stalk crinoid might be in the genus Pneumacocrinus. But Pneumacocrinus usually has long filaments on the ends of its feather-like arms. And I didn't see filaments on this one, so I'm not sure. It might be a Bathycrinus. There are a couple options. Of course, I'm not a crinoid expert.
I'm guessing the Bathycrinus also lives on bathymetry. Would that be correct?
That would be correct.
All right, thank you. 2 for 2.
2 for 2. The geologist is getting into it.
Did you snap zoom?
Okay.
Oh, what do we have here? Is this a stalk of a sponge with hydroids on it?
Yeah, it looks like a dead stalk of a sponge.
Oh, we're seeing them all over. And anemones, a couple
Oh, we
anemones.
saw this yesterday where
and,
there was a...
something here, too. This,
Yeah, a little clinger on there. So yesterday we saw a sponge stalk with cereanthids on top, and I think this is the same thing.
Did you need a closer look at that, Megan? Or?
No, we don't. I don't know if we need that. We can zoom in on it later.
Now let's move on up.
So that guy was probably a polychaete or maybe another benthic ctenophore. But most of our associates that are looking, or most of our scientists that are looking into the associates living on corals and sponges are looking for associates living on live corals and sponges. But it's also very interesting to note that even dead stalks of sponges and dead skeletons of corals can be homes to animals. So this is some really interesting geology we're seeing here. Some like lava flows?
Yeah, I believe they're lava flows. It gets a little hard to tell when you have all the thick coating of this manganese crust on top. It mutes the shape and the edges, so everything kind of looks smooth and sometimes shiny, and you kind of lose the ability to make a good good identification of it. But what we know about the geology and the context of the area, I'm pretty certain there'd be lava flows, and then this talus, this broken material, broken rock in between where they've disintegrated, degraded over time.
Okay, you want to do a snap zoom? I think it's that same coral, but let's see. Maybe the polyps are closed up. Okay.
Look at the curved rock structure.
So Tina said in the chat log that it might be the only way to collect deep sea cereanthids is through this. Oh, you turned on the mic.
Sorry, got the wrong button.
Is through collecting this dead sponge stock, but
We're about
since
ready.
we've seen this before, maybe we'll see it again.
You got your mic on. Turn the green off. Mute your mic.
Sorry.
Do another move.
Oh.
All right.
Okay. Got it.
Yeah, we can put in another move. Let's adjust to 100. We'll do another 30 meters bearing 100, 2/10. If Dan's comfortable with 2/10 still.
Copy that.
Up there.
To confirm, you said 2/10.
Yes.
Yep.
0.2.
The white coral looks different. It's got longer polyps.
Bridge ROV nav.
Go ahead, RV nav.
We'd like to put in a ship move, please.
Okay.
30 meters, bearing 105.
Let's do
0.2 knots, please.
Is that 30 meters bearing 105 speed decimal 2?
Correct.
Okay.
Thank you.
I have 100 on that there.
Oh, bridge. Belay that. Could we make that bearing 100, please?
ROV nav. You said change that to 100.
Go ahead and zoom in. Roll.
Affirmative.
Okay, so that's 30 meters bearing 100 speed decimal 2.
Affirmative.
Okay.
Thank you.
Okay, so we are seeing a chrysogorgid coral right now. This is a very deep sea chrysogorgid species. Oh, and my computer just made the sad blue screen of death at me.
So what's unique about these corals versus the other ones we'll see?
Well,
anything?
One thing is, you notice all the polyps are again all on the same side, and it's branching. So it's not like the chrysogorgids we were seeing yesterday that were spiraling like Iridogorgia. These might be
See the current just whipping by.
Chrysogorgia.
Ready for full screen?
Go ahead.
I know we collected a couple deep sea Chrysogorgids in the past. I'm not sure. Is anybody saying anything?
Oh,
Plurogorgia?
Plurogorgia.
Yeah, it looks
Scott,
relatively
Tina.
young.
A juvenile.
Okay, video's clear.
Okay, zoom out.
Yeah, so it's really great to have these onshore scientists participating. That way, you know, we have more input as to what we're seeing. They have more ideas about what to collect and what's important, how to ID them. Because no one person can be an expert in everything.
Hard to be.
So I think we're seeing another Anthomastus or mushroom coral. Is that right, Tina?
Go ahead and zoom in.
Looks like he's missing a few of his appendages on the bottom there.
Yeah, so Tina agrees that this is probably an Anthomastus.
Okay, go back into one.
Has very long polyps, very long tentacles. So these are a type of soft coral. And I really like them because when you collect them, they pull their polyps all the way inside their body and they look like little mushroom from Mario.
So with these ones on the right side of it that are shorter, are they, would those have been predated upon and grown back, or are they just...
Yeah, I think this one polyp that's shorter over here got nibbled on at some point. He's definitely damaged and regrowing tentacles. That little polyp got.
Video's clear.
Okay, clear.
Are there usually just 4 coming out, or they have multiple?
They can have a lot more polyps than that.
Okay.
And it could just be that this species has fewer and larger polyps, or maybe it's a young one. I'm not really sure. Wow. Oh.
Get too much further ahead of you, Jeff.
You got room.
Yeah.
And we're moving.
Associate on the coral to the left.
Oh, can we zoom in on this sponge sock?
Sure.
Let's see if it has cereanthids on it. It definitely has some sort of brittle star
Okay, go ahead.
wrapped around. Let's go to the top. Not sure if those are cereanthids. But there are some sort of cnidarian up there at the top. Guys in the chat room, any opinions?
Got more zoom, or is that max?
It's max.
Okay.
These ones don't look as nice as the ones that we saw earlier.
Oh.
Oh wow,
Okay.
that was a lot of
Yeah.
backlog.
Okay. Zoom out.
Has anybody said something, John?
Yeah, I think
I have to relog
she
into my
wanted to look
pigeon.
from the other side. Yeah.
Is there a way to see the other side of that stalk before we move on?
Sure. The right side?
Yeah, where the larger polyp was, like pretty much the opposite side.
Oh. Well,
if not, that's
I can
okay.
I agree with Roland. I would say left side.
Got it. Okay.
That top left anemone looking thing almost has a tube.
I'm not connected
ROV
to
nav,
the
bridge
internet.
move complete.
Copy. Thank you, Bridge. Move complete, pilots.
Thank you, Nav.
No problem. And just to confirm, we've been moving at a bearing of 105, 100. Are we going to continue to swing a little further to the north?
Yeah, I'm just going to follow the contour best I can. So basically, we're
Okay,
looking
go
at the
ahead, Roland.
sonar. I'll get out of your ear, sorry, pilot.
You're right.
Got the sonar, you can see this sort of larger ball of yellow here. So I'm basically
All
just
right.
lining up my heading
Sure.
so that's facing the middle
We are
of that,
zooming
assuming
in
that's
on a
the
different
most,
angle.
like local high. And then wherever I'm facing when I'm lined up with it is
It
the
looks
direction
like
we'll go. So you can see right now I'm like
there
almost
might-
190
there's a number
or
of
090 or
polyps
085
on the top
so
of this sponge
we'll end up coming
stalk.
around a little bit more.
Copy that.
I can't see if there's that inside circle of tentacles that are
Slipping.
typical of cereanthids.
And co-pilot, I'm sure you've seen, based on the high pack, it looks
Oh,
like our
okay.
bearing to hit waypoint 2 would be about 77
again?
degrees or so, 75.
So it's our hunch
Okay.
that it's probably not
Yeah,
a
so we'll keep-
cereanthid.
We'll keep crawling around to that, it seems.
Copy that. Sounds good.
But yeah, we generally just follow local high for the ease of the pilot.
Makes sense.
And it keeps the slope in front of you, which keeps the view interesting.
So I'm thinking we should probably move on.
Yeah,
Okay.
I think we should
All right, let's zoom out.
get moving on.
All right.
No
But if we see
consensus
another
here.
one of these, we should zoom in and see if we have those cereanthids on top.
All right, Jeff, you should hop in.
Say again?
You want to hop in?
Oh, sure. Want to push out first? Or?
Yeah.
So the comment by Scott was that they look like they're probably small anemones rather than these cereanthids.
The current's going to blow you away pretty quick. Thanks, Roland.
So if you're just joining us today, we are coming to you live from the Okeanos Explorer, and today we are diving on Beethoven Ridge. We are currently at a depth of about 2,485 meters. And we are looking at what kind of geology are we seeing, John?
Well, we're looking at volcanic material. That's kind of what we're looking at right now is a combination of intact lava flows and broken rock or talus, or also called scree.
Left edge of screen going towards the bottom.
That's with us. You can tell that part of it because it has this light-colored material, which is the sediment in between it. More so than the other flows, and that's all coated by this polymetallic crust. So it all kind of smooths out the look. We have a nice zoom of it now. Makes it kind of a bumpy texture.
So I've been told that this crust might be valuable. Why is it valuable?
There's all kinds of metals and minerals in it that might be of interest. There's iron, there's manganese, cobalt, rare earth elements, and all those things are used in our modern society for electronics, solar panels, cell phones, all kinds of things used in aircraft wings.
Yeah. Yep.
Copy that.
That's really cool. As for biology, so far we've seen some pretty interesting things. Some black corals, some primnoid corals, sponges.
Put in another ship move, please. Okay.
We saw a Goni sea star.
100 at 0.2 knots, please.
Actually, we've got a little different coral coming up.
Yes, let's definitely zoom on this. This is definitely different.
Right.
Let me get around. Hopefully, with the current, it'll allow me to get around the fan side here. So we can get a profile view. Not usually.
ROV nav bridge move of 30 meters, bearing 100, speed decimal 2 has been initiated.
Copy, thank you, Bridge.
You're welcome.
Ship move underway, pilots.
Thank you.
Starting from here, video.
Okay, Jeff, you need to lift your mic up a little bit.
In a little bit. I can't do it right this second.
Yeah.
All right. Well, this looks to be like a corallium, or a corallidy coral, or a pink coral, or red coral, depending on, how you'd like to give its common name. This is likely the genus Hemicorallium.
Approximately 4 minutes or so, 3 to 4 minutes.
Okay.
I'm sorry?
Any part you want to see in particular?
Can we see the top of one of these branches?
We'll zoom in closer on one of the ends, sure.
Max zoom.
Max zoom, okay. I can fly in a little closer then. You want to come out. It's just a good perch for a profile view. Get in real close for a view of the ends here.
So this is a pretty deep living species and genus.
Okay. Forward.
I'm not sure if I can, if I were to make a distinction of what species I think this might be, it could be Hemicorallium bizale. And these are relatively deep living species, tends to not have as dense of a branching pattern. And we see that the top of each one of these branches are topped with 2 polyps. And we've seen Hemicorallium bizale in the Northwest Hawaiian Islands. So if this is indeed the same species, there might be some connectivity between the Northwest Hawaiian Islands and our musician seamounts.
All right, well, that's one of the goals of the mission and the dives to determine if there is or isn't.
Yeah. Oh, look at the bottom. Would be really nice looking at that.
Is there a specific
Yeah, there's a good
distinguishing
polyp lower
characteristic
left.
you're looking for here, other than the
Shows
color
deal
and?
that she's talking about.
Well, the color usually gives it away as
Okay,
to what
definitely moving
species
now.
and or
Yeah.
as to which family. The corallium and or the corallidae
Oh,
and the
There.
Paragorgidae tend to have that very distinctive pink color. Now, that's not always true. There are some colonies that I've seen that are white. There are some that I've seen that are a lot darker pink or darker red. But for this Bizale, Chris Kelly was telling me that the best way to sort of get a sense of if it might be that species is if it has the not very dense branching pattern, and it definitely has these two distinct polyps at the end of each branch.
A little ways off. Oh, there.
Okay.
Nope.
Thanks.
But that is a different one.
Oh yeah, let's zoom in on that black coral to the right of the screen. That's definitely something new. I think it looks like it's branching.
Coral or the white black coral?
The orange-looking one, the very fuzzy one. So I find black corals also pretty difficult to identify, but fortunately we have some onshore experts. So black corals are hexacorals, meaning they have 6 tentacles on each one of their polyps.
Is that why they're called hexacorals?
That's why they're called hexacorals.
Amazing.
Maybe this is Trisopathes? Let's see.
Where would you like
Maybe
to look to
Tina
help you
knows.
sort it out?
Go for full screen.
Copy.
ROV nav bridge move complete.
Copy. Thank you, Bridge.
You're welcome.
Well, it's a very
complete
beautiful
that.
colony.
Looking down at you at 80.
Thanks, co-pilot.
Video's clear.
Pilot's clear.
There's a dead sponge stock. Can we take a quick snap zoom on the top there to see if there's any Cereanthids, pilot?
Yep. It will have to be a snap because I'm
Yeah.
trying to keep up with the ship, but we will get it.
Now these ones
towards
actually
the
do
right.
kind of look like the Cereanthids. We might need to collect this.
Okay, easy stop on the ship.
Yeah, it's already stopped, but I've got another 3 minutes of swing or so. We can back up if you want to do that.
You want a tighter look to decide whether to collect?
Yes, please.
You can judge him. Judge
Yeah.
it, I'm not sure.
Yeah, why don't we
So
go
sure,
180
we're seeing
where
another
those other
dead
moves were.
sponge
So
stock
we're gonna move
with,
back.
I'm fairly
Move
certain this
back,
time that these are the Cereanthids
10
we saw
meters.
earlier.
10 meters.
Yeah.
Copy that.
I've asked the ROV team to bring the ship to a stop, and I think we might collect.
Bridge, ROV nav.
Go ahead, RV nav.
They don't look as translucent as the ones we saw before.
A few meters. So we'll
So
go.
run away.
We'd like to move the ship 10 meters at bearing 280 at 0.2 knots, please. 0.2.
Okay, 10 meters.
But of course, in order to confirm that these are indeed Cereanthids, we need to see inside the
Affirmative.
center of the
Thank you, Bruce.
polyp to see if there's another
Okay.
ring of tentacles
Rotate 90 degrees.
that are characteristic of Cereanthids.
Around, look inside.
Your audio is still very low.
So why all the interest in these?
Well, they generally settle down into the sediment, which makes them very hard to collect, especially in between rocks and stuff. But when they're on a stalk like this, we can easily pluck it with the manipulator arm. So this might be the only way to collect this type of specimen, especially without damaging it.
How tight of a zoom do you need
Would
to get
they
into the
just
center?
jump out of the stalk? And
We
Looking
need
for
I think, a
additional
pretty
more
tight
tentacles.
zoom. We just need to see if there's tentacles on the inside. So I think we have a good view right now if we can snap
in.
zoom.
RV nav bridge.
Go ahead, Bridge.
Hey, we were just ramping up a little bit, so I had to decrease the speed to decimal 1. I wanted to just let
Does
you
it
know.
look like a second ring of tentacles
that,
to you,
thank you.
John?
I see some smaller ones in
I
there.
see
I
some
don't know
smaller
if you
tentacles
call
on the
that
inside.
a ring or not.
Pilot's the bridge informed me
That's
that they
one
have
of the
to
smaller
move it 0.1
ones, I believe.
knots.
Yeah.
Copy.
Ah.
Shoreside. Scott, Tina, Asako. What do you think? Yeah, I see sort of shorter ones on the inside of the mouth, so it looks like it. So
the
pilot,
lower
we'd
one
like to
looks
collect
like a
this.
It's max soon.
Sorry, focus on one.
Scott gives us a go for collection.
All right. Come back around to a better foothold. For ease of collection.
This is pretty
Ship is
exciting.
backing up.
Being able to collect a Cereanthid
Still waiting on that.
intact
That move,
inside
though.
a
It's looking
sponge
little
stock.
close right now.
All right.
We don't really have a lot of specimens of these deep sea Cereanthids because they're very fragile.
Grab and go then.
Probably another minute or so until you feel that move, co-pilot.
I think we'll
So,
be all right,
shoreside,
Jeff,
what would
but-
be the best way to preserve this specimen? In formalin? Or should we do one in formalin, one in alcohol?
Yeah, I think you'll be all right, Jeff. I'm just staying pretty high above you, and you might feel a little tether tug, but
In pilot,
maybe not.
can we try to get the whole sponge stock?
So try to pull the holdfast off or cut as low as possible.
Yeah, well, as low as we can, just in case there's anything else inside that stock.
Okay. I'll try to grab it with the fingers and kind of low and
just pluck it off.
Pluck it off.
Watch out for that current.
The current.
If it gets loose, it's going to blow away.
Oh yes, that's true. Video is saying if we cut it, we'll have the soft grips, and I can just firmly hold it with our soft jaws. Versus if I use the fingers, it will take some skill to hold
Looks
it
like you may be
softly.
starting to move a little bit, co-pilot.
Okay.
So do you want to risk me possibly dropping it or not is the question.
Okay.
I think video's trying to say.
ROV nav, bridge. Move is just about complete.
Copy, thank you, Bridge.
You're welcome.
Move is coming to
spun
completion,
up?
pilots.
Yep, spun up. And
Also looks like a
everything's
glass
ready to
sponge,
go.
which makes it
I
brittle.
assume the port bio for you, but
Okay, arms coming live. Okay, we'll reach out here. Okay, let's get the minis just up here and lined up.
all right. You got it.
Either that or the, I don't know if I'll be able to see the mini. Maybe we'll have to bring up the pilot, the Titan. See what I can do. Yeah, that's probably better. Let's move it up a little.
Okay.
Just get the,
Sure.
those two. There he is. Okay.
Can you help me keep an eye on the Sirius camera, John?
On it.
Thanks.
What angle is your Sirius cam at, co-pilot?
It's 101.
Okay,
So we're kind of looking backward a bit,
gotcha.
but we're definitely moving
That's good.
back towards D2 now.
Bring it back into view a little bit, yeah.
Yeah.
force 2, I think. I'm going to try to grab it. Okay. Come up slow. Okay. Let's try to go for the port side boxes. You think we're able to open the drawer?
Let's see if we can. Nice and slow.
The left arm is bumping it as it goes.
Oh, that's it.
It's
Gonna have
not
to come
enough.
up. Yep.
This is a lot of current.
I'm going to retract just a bit.
Okay.
Just so we don't scrape the wing. There we go.
Did you guys copy that the left arm is rubbing against the drawer?
Thank you. Okay, we're going to have to pick up and fly shortly here to be able to open the boxes. So bear with us for one moment.
Okay, so we've picked up our sample. Right now, pilot's getting into position to open our bio boxes.
Co-pilot D2 is slipping off the left side.
So
of
in
the
just a
camera
moment,
there.
you'll see those boxes move out
He's just picking up
and
sit down again so we
open.
can get the drawer open.
Copy that.
Steady enough for now. Okay, if you want to
Ready?
open the drawer, yeah.
Okay. Fully open. Do you want your mini Zeus back, or you want me to follow you with Titan or switch over to mini Zeus?
You can switch to mini Zeus.
Okay.
Okay, I'll try to delicately drop it in. And it's in the box.
Closing it
It's
up.
in the box. Great job, pilot.
Yeah, I see it.
All right, that
I'm gonna
means
move the
that I
shilling
can
back out a bit.
collect. It's official. We have collected the specimen.
Nice work, pilot.
All right, arms off. Do you know to drop the target and all for that?
Yep, I already have that marked on HiPAC. That's specimen 2 bio in port outer box.
All right, let's get your line back up.
Onward and upward.
Wonderful. So now that we've collected, it's super exciting to have these Cereanthids be able to preserve them. So right now we're having a little mini conversation in the chat log about how to preserve the specimens. So it looks like we had
It's
at
like
least
a very
3
large barnacle.
polyps of Cereanthids. Tina's saying that they might be in the family Arachnactidae.
Got your toe in Sirius.
And we're going to possibly preserve 2 polyps in formalin, and that'll allow us to see the different structures inside the polyps, and then 2 in alcohol, or 1 in alcohol, and get genetics. So I'm seeing another Anthomastus, Bathycrinid, 2 Anthomastus. So those Anthomastus are the mushroom corals. These ones seem to have very long polyps.
Got you a comfortable 60 degrees.
Great. Okay, watch lead, we're back on track, so if there's anything of interest that you want to see, we're back to normal operations.
Looks like it is starting to curve to the north, which is what we expect from HiPAC.
Yep.
So pink thing on that coral, big rock on the lower left.
You see the fuzzy right over there now.
Yes, let's zoom in on that little fuzzy organism on that rock.
Yep. Trying to find a good perch nearby, and we'll get a good zoom on it for you.
How's our progress, Nav?
It's got 2 planes to it, it looks like.
Looking good so far. We need to average about half a knot or so. I'm going to make an update.
Half a knot?
Correct.
Or
I'm sorry, half
a tenth?
Yeah, half a
knot?
tenth.
Okay.
Apologies. Yeah. Yeah, so 0.05 knots. I'm going to do an update now that we've backtracked a little bit.
Okay.
Come on in.
So from this angle, when we're zooming in, we have a chrysogorgid coral. And there seems to be an associate on one of the branches. Appears to be a squat lobster. You can see his claws. So a really cool thing about lobsters and crabs is that the males tend to have the more robust claws in a lot of species, and the females will have less robust claws. So I'm not sure which one this is. It might be
Can you take your camera?
a Uroptychus.
Nope.
Okay, I'll just pump up a little here and see.
Copilot naps.
Go ahead.
We still, have about
And this might be
230
another
minutes of bottom time left and need to maintain.
Scott is saying
slightly over,
the skeleton is
0.05
gold,
knots.
so it might be a Chrysogorgia. So in the genus Chrysogorgia.
If we were to make a direct
Asako
full
is
screen
saying
waypoint
Plurigorgia,
to,
so there's a little,
approximately
you know, full species, please.
374 meters to go.
Okay.
So it's hard to tell. I think we're actually seeing 2 separate colonies.
I think this left part is a
that
branch
are right
off
next
of
to
it.
each other.
Well, I can spin around and get a look at the holdfast, but it looks like it's branching right there.
Oh, is it branching right in the middle?
Yeah, right behind, or right off the back of the squat lobster. It looks like the branch to all this is.
Oh, that's very interesting. It looks like a
different,
double
side
planar.
view.
You could try.
Then,
Yeah, I just didn't want to lose this
yeah.
perspective. Pointing it out. I could get to the right side of
Yeah,
it.
that's the best view.
Okay, come around to the right. Maria. It's going to darken it a little with the rocks, but that's okay. Make for kind of a cool view. Alright, come on in.
Well, that'll give us a good view of the squat lobster.
I only see one holdfast in there.
Yeah, only one holdfast, so it's a chrysogorgid with 2 parallel fans. And this squat lobster does look like Europtychus to me, is in the family Chirostylidae. The Chirostylids are known for having these long, slender arms. And he has a very short rostrum, and that's the little projection between his eyes. And this little creature might be an amphipod, so a different type of crustacean.
Please pardon the interruption. Your conference contains less than 3 participants at this time. If you would like to continue, press star 1 now, or the conference will be terminated.
I'm the only participant. Oh my goodness.
Full of this perspective.
Yeah.
And then move on.
But it looks like a pretty large amphipod to me. Amphipods, in most cases, tend to be relatively small, but in the deep sea, they've been known to get to be pretty large. That's some good imagery from science. Of course, if we were to try to collect a coral like this, most of the time these little crustaceans will bail. They're pretty smart, and I just, the way the squat lobsters swim is great.
Alright, you want to come wide?
Yep, they just sort of do this little puttering, little squish type of move. They open their tail and close it really fast to sort of zoom backwards. I don't know if I'd feel comfortable swimming backwards.
Oh no, just in my other camera. Like
Well, at one point there were a lot of sponges in this area. We've seen a number of dead sponge stalks. Here coming to view is another dead sponge stalk that's, you know, coated with some brittle stars. They're just all wrapped around the stalk.
Do you want to range down to 20 meters on D2?
Do nice side to side here.
Copy that.
Rotate.
Down to 20 meters.
Nice. And then
also let me know.
Yeah, I don't think we're quite there yet.
The new slope, it's a little bit
But
steeper now.
the next move will be at 100.
Copy that.
So about 45-degree slope now.
Nav, watch lead.
Go ahead, watch lead.
How far are we along our transect?
We started at about 500 meters from waypoint 2. We're approximately
I'll get a movement when he's done.
375 meters out at the present time. So about a quarter of the way.
Thank you.
All right, Nav.
No problem.
Now facing towards the south.
Let's go ahead and get a move in toward 100.
Pink thing on the upper left.
Ooh, a fish.
Fish.
Copy that, go by
There's
it.
frogfish. There's an eel at the bottom right.
We'd like to go another 30 meters at bearing 100.
You can snap in on the
I think
fish.
that's fine, yeah.
Copy that.
Oh, we saw this fish earlier?
Bridge, ROV nav.
He followed us up.
We'd
Yeah.
like to do our next ship move, please.
He looks almost
We'd
like the
to
same
move
one.
30 meters.
Similar sky.
Bearing
this is
100
a
at 0.2
Ileophus.
knots, please.
It is a type of eel.
Speed decimal 2.
And
Affirmative.
Bruce was telling
Thank
us
you.
yesterday that its common name is the Aerotooth eel. So Bruce Mundy is our fish expert over in the Pacific Islands Fisheries Center, located on Ford Island in Honolulu. So these Ileophus eels, they have a blunter nose than the other subfamily of
Copy that.
eels.
Thank you, Bridge.
I think he's getting a little
Keep going up,
confused
you're
by
gonna
our
get that
lights and running
toadfish.
into the rocks.
Say it again, Roland.
I think if you kept going up, you'd hit that toadfish.
Come on out.
Another thing to look for when identifying
Straight
this
up.
eel is along the side, there's a lateral line of pores,
See the
and
red
those
fish?
are
Toadfish.
white pores. They're very distinctive. They also have a
Down and
longer
come a little closer,
dorsal that
Roland.
starts
Yeah,
earlier
get as
up on the
close to it
body.
as you can.
Okay.
We're seeing another
You want a profile
goosefish.
sight?
Straight on the way you got
My
it.
personal now favorite. This, the third time we've seen this fish. So every dive so far we have seen this fish. This is Chaunacops coloratus. It is in the anglerfish family. And as you can see, there's like a little white ball in between the eyes,
Little
and
camera
that's
tilt
the
down.
lure. They use this lure to attract prey towards its mouth.
Come on down.
Think
Come
you're
on in.
getting closer.
I know, it's a very small one. The one we saw yesterday was a lot
I didn't want to speak
different.
from it.
It was a darker, deeper red,
Run away when I was.
and it was a lot
Yeah.
larger.
Okay, that's good.
So there's some discussion right now between our fish biologists whether the fish that we saw yesterday that was of a deeper red color is in fact the same fishes we're seeing right now. I'm seeing these long fleshy protuberances along the body just like the one yesterday had. That's a beautiful zoom. You can really see the lure. And to a shrimp, an unsuspecting crustacean, that must look like a delicious
Slight
treat.
tilt down.
Slight tilt down.
Oh yeah, it does look like a little
Like so.
Dr. Seuss tree. And look at that mouth. It's so neat. This one hasn't been phased yet by our staring at him and hasn't swam away. But the first time we saw this fish, it did swim away, and you could see
Looks like I'm on the
the gill vents, and those are actually located near the tail of this fish, which I
Pulling
didn't
out.
know. Usually, with most fishes, you see the gill slits right behind the eye,
up.
but in fact, they're behind the leg-like fins. Lovely. I think we're good for science. If video is happy.
Video's clear.
Pilot's clear.
Another big coral up ahead of you, Jeff.
See that?
Video swapping out.
Thanks, Roland.
Oh wow! Let's check
Big
out this
drill.
black coral.
All right.
This is very interesting. It looks like a Bathypathes that has decided to branch sideways.
Try to get the branch point.
Maybe Tina and Asako might be able to help us out with what's going on here. This is very interesting.
Okay, video, you can zoom in. Going first thing for that branch point. Well, when you're settled. They just did a video swap out. We have Bob in the seat now.
That is really, really interesting. I mean, it looks almost exactly like the one we collected yesterday, other than the fact that for some reason it's branching off to the side, and I've never seen a Bathypathes do that. I'm not really sure why it would start branching this way, but this branch point is really odd to me. It looks like some of the branches on the branched point is also branching. So I'm not sure if it got damaged and it started growing in the wrong direction, or this is something we've never seen before. Tina Molitsova is saying that sometimes Bathypathes does do this.
Want to get a specimen view? Video?
Yeah, can we get a whole specimen view? Yeah, Tina says that when the main axis is damaged, sometimes they will branch off. So, but it does look very similar to the one we collected yesterday, so when we have good imagery of this, we can move on.
Pilot's clear when you are, video.
Okay.
Awesome.
Got you at a comfortable 55 degrees.
I'm going to do a short little drift test here. I think the current has calmed down at this depth.
Okay.
I'm looking at 2,450 meters. And I guess it really hasn't calmed down. It's just switched to my face. But the current's at least shifted, and it possibly is also slowed down.
Well, that's good. It seems the current is shifted and slowed a bit. That should make our transit a little bit easier. I'm really hoping to make it to the top of this cone feature.
Stock or something up to the upper right on Sirius.
I've also been seeing a number of little fan-looking corals, so when we see some of those, let's zoom in.
Something real
large. And there
large.
is another fish to the right. It's probably it looks almost like the same eel.
Back for more.
And then I really like to get a view of this coral dead ahead. It looks like a hemicorallium to me.
Okay.
ROV nav, Rich.
You're okay with
Move is
this
just about complete.
this fish? You know what it is?
Yeah, we're good with this fish.
Ship move is coming to completion, pilots.
We're going to come to this large boulder. This looks like the underside, so I'm going to see if I have enough tether to get around to the polyp side.
I think you do.
Yeah, the really interesting thing about these corals is that they like to face the current. So the way that each one of these coral fans is facing will give us some interesting insight into which way the current generally flows in this area. There's also a squat lobster, it looks like, on the backside of this coral. And as we're getting closer, I'm fairly certain that this is hemicorallium. It's quite a large colony. This genus grows pretty slow. Something like this might be upwards of 100 years old. And this is different from the one we saw earlier. You see how much more dense the polyps are. So this is likely a different species than the one we saw earlier in the dive.
And you were interested in the crab?
Wow, that's pretty incredible.
Welcome back, John. How was
Thanks.
lunch?
It was good. Pizza Saturday.
Oh, Pizza Saturday.
This crab.
That's exciting. Let's get this crab, and then I'd also like to look at that fuzzy coral off to the right,
one, looking one, or?
that, V-shaped coral.
Yep.
And this looks like possibly another Munidopsis? No, maybe? Munidopsis? Look at that, is a very long rostrum on this squat lobster. It's curving upwards, so you can see it looks like a giant rhinoceros horn, and it has a number of spikes on it. So the shrimps and also have this type of rostrum, and when we want to ID shrimps, it's important to count how many spines. And for a lot of lobsters and crabs, that character can also be quite helpful in identifications.
What do they use that for?
I'm not sure what they use it for. It tends to be highly variable across genuses and species, even family. Some have little to no rostrum, like the squat lobster we saw earlier has only just a tiny little projection, and then others have an extremely large rostrum.
Some black background for it. Come on in.
So this is a primnoid. You can see it moving around in the current, likely Neurella. Yeah, rock. John, did you notice how this rock that the corals are on is really shiny? Does that mean it's newer, or is that something that the manganese crusting does over time?
Yeah, it's not that I know of that necessarily anything newer than any others. It's just for some reason certain parts of the crust are kind of shiny, or they look like they're polished. I don't really know why. Excuse me, I don't really know why that is. It looks fairly thick on this outcrop, the crusting, and when I first came in, I noticed this feature was looks like a big
get out of here.
Extrusion of lava. So it's kind of an interesting outcrop. I like to take a look at it once we move away.
Up to the peak.
All right.
All right. Good for science on this coral.
Cool. Let's get out of here then and keep up with the train.
All right. Lasers back on. And,
What bearing would you like for the next move, pilots?
Let's let D2 get out a little bit and help us acquire the slope.
Yeah, but you are going to want to come around.
Yep.
Still, so I'll sidle port a little further before I push out.
Yep.
All right, Megan's heading out the door to get some midday meal, and hopefully keep you entertained with some of the geology and along the way, and see what we can do with some of our onshore scientists as far as the biology as we go.
So I'm heading like 075 right now.
This looks like a pen that we haven't seen yet. I know that. See if the scientists on shore. We'll take a snap and see if they're interested in looking longer. Yeah, come on in. A little rock pen of sorts. Something like that. Let's try brakes. Snaps it in. Yeah, I don't think we've seen this yet. Are you getting any interest on this? Or should we continue on?
I think we normal Said we had some, Tina says this is a normal pen. I was going to say, thought it might be a C pen.
Okay.
I think we can go ahead and move along.
Okay.
Doesn't seem to be a large interest in this. A number of items in the back, but I think we can continue on the track.
Looking at maybe a 0, like a 070. What does high pack estimate?
Let me get an updated reading for you here. According to high pack.
So we're moving up
We would need a heading
another
of
slope
approximately
that's mostly talus. This is all the broken rock from this, in this case, from lava flows upslope and around this area, and some really large pieces here that we're seeing fractured. This kind of angular or prismatic morphology
just ready, we can go
that
do
you
it.
can see.
30 meters at 0.2 again?
Yep.
Copy that. Bridge ROV nav.
Go ahead, RFE nav.
We would like to put in another ship move.
Okay.
30 meters. Bearing 070.
It's kind of unstable surface, as you can imagine, so it doesn't provide a lot of long-term stability or good substrate for large coral colonies or sponges to grow and evolve.
Camera's at 30.
Because it might be continually moving downslope or other rocks coming down, tumbling on top of them. So that's one reason that we're likely not seeing really large biological features attached to these rocks in this area. So the other couple dives before, we saw a good example of where there was a really big lava outcrop that was intact, and then surrounding it were all this kind of broken rock, this talus. And there wasn't much on the rock, but on the broken rock, the talus, but on there were a number of colonies on top of the intact lava flows that you could see. All right, so it looks like we have a little sea star here.
It's on a stock sponge. Knocked it right over.
Oh, it's on a the
sponge holds fastest at the top, and it's just falling over on the rock.
Yeah, it's like this.
Can't hold the weight of the star.
Yeah, there's a comment in the chat room about the sea star's kind of full. He's pretty much eaten away at this thing that he's grabbed hold of.
Okay, I think we got plenty of that. Continue on.
All right.
A minute or two before serious moves.
Okay. We can move along. So Scott said it may have been eating on a black coral, but it's difficult to tell because he's pretty much eaten everything away, and it's a thin skeleton. And there was looked like a snail on the end of that near the stalk.
I wouldn't go too much further out ahead yet.
This might be Scleractinian. Or at least some sort of coral or anemone. Come on in, video. I'm at the end of my leash, so I have a little time, so I don't think we've seen one of these yet today, so I thought I'd take a second.
So we have this is a cup coral of some of our shoreside scientists have passed on the identification. I guess it's a single cup.
Okay, I think I'm on the move now.
Great.
And it's the first Scleractinian coral that of the cruise, according to our scientists on shore, that we've seen.
All right, we've got another second, and we'll continue on.
All right, I think we can move along, pilots.
Cool.
I'm told by Scott that these are hexacorals, so they're more closely related to anemones and black corals than to the octocorals. And yeah, to me they look quite a bit like sea anemones. So for those of you that have just recently joined us, this is the Okeanos Explorer, the NOAA ship Okeanos Explorer, coming to you live with some delay from the Musician Seamounts. And we are currently operating a two-bodied, remotely operated vehicle, a robotic submersible that's down on the seafloor, about 2,400 meters down.
Heading got pulled off a little bit. Coming back. 070.
Looking good there, pilots, and I think we're going to leave the next shift in good shape, making good time.
Awesome.
Cup coral down there has got its polyps closed.
So now we're moving into some larger rock outcrops, some bigger, much larger, more pronounced lava features.
There's a group of fungi.
And basically probably a more stable surface for a lot of these organisms. So we may be seeing some larger colonies or just larger stalked sponges as we move up, things that are older and been around longer. These rock forms look like fractured pillows or lava lobes that you're seeing in front of us, and that probably contributed to the talus or the broken rock that's down slope from what we've been traversing until now. These are well coated with this polymetallic crust that I've described earlier. It coats the features, it precipitates out of seawater at a very slow rate.
Try to make it.
But over millions and tens of millions of years, it can build up and kind of mute the features, mute the edges, so everything looks kind of rounded. And it gets a little bit difficult to identify sometimes as the crust grows.
This is unfortunately dead, but it looks a little different than what we've been seeing.
So this looks like a dead sponge stalk in front of us.
So that's probably about the end of our move right now in terms of what we're feeling.
I believe,
about 5 minutes, and we felt it at about 50.
Correct, yeah. I think the bridge will be calling me in just a second. And you should continue moving
Well,
for another...
I think the ship's already stopped, but I think even Sirius is not going to feel it after a minute or two now.
Yeah, I would expect you still got
Looks like we have a couple dead sponges lying around the rocks. I think that's a fairly common thing you see at the base of a slope change. These sponges are growing upslope, die off, tend to tumble down over time and collect in kind of crevices and parts of the rock that collect them kind of like a... Sometimes we've seen kind of a whole collection in a sponge reservoir, so to speak.
Try like a...
See, now we're moving into
we're gonna have to shift
more
again
organisms as the slope increases upslope. We get some big intact features. These are all lobes of lava flows, pillow lavas.
Let's get another move in.
And these things have been intact and around for a while.
Would you like to put another move in?
Yes.
During the shift change?
Yeah, well...
Okay.
Yeah, I think it's okay.
Same move as last one?
Yeah.
Copy that. Bridge ROV nav.
I'm sorry, John. No, 060.
Go ahead, RV nav.
Copy that, co-pilot. We'd like to put another ship move in, please.
Sure.
30 meters, bearing 060 at 0.2 knots, please.
That's 30 meters.
You're out pretty far.
060 at 0.2 knots.
That's affirmative.
Interesting.
Thank you.
We're on like a nose. I can't venture left and right too much without going down.
Yeah.
It's a ridge.
Big stocked sponge in the background. Yeah, you see it too.
So a lot of these white, these small white coral colonies are told are Primnoids, and we see quite a few of those now as we're moving along this new part of the seafloor.
Ship move underway, pilots.
Have another sponge coming into view. Stalk sponge.
Something. I'm interested in knowing what is on the surface of this rock.
RV nav bridge.
Go ahead, bridge.
Hey, just a question. Are we going
Probably
to
could we take a look at this rock right here in
kind of
front of us and see this fuzzy stuff growing on top of it? Can you take a zoom on that?
Yes, that's affirmative. We've finally kind of settled on the direction we're trying to achieve. It'll probably be in the 060 to 080 direction from here on out.
Okay, great. Thank you.
No problem.
All right, I do not know what this is. So
These are carnivorous sponge. I made that up. That's my guess.
kind of looks like lichen on a rock on land.
I'm just going to touch down here. Get a little bit more stable shot. Let's have some interest.
So, hydrozoans, it sounds like. Oh, yeah, we kind of have a debate going on here in the chat room. Hydrozoan colony or cnidarians, polyps being visible. Ah, I see.
You need more time on
No
this?
debate. It's there are hydrozoans are cnidarians, so
Video.
All right, pilots, I think we can move along.
Now, co-pilot.
Hi there, co-pilot.
When did we put that last move in?
Last move was initiated at 21:57.
Copy.
So we probably have another 4-ish minutes of movement. You should be feeling the movement of the ship right now.
Yeah, I think I am moving forward.
Copy that.
Was that at 060 now?
That's affirmative. And we should be heading in this general direction for the remainder of the dive.
So
terrain permitting.
Sounds like that.
In the 060.
It was a bit odd that we
range.
had all these hydrozoans on a single rock, kind of collected on one mass. So there's something unique there. All right, we have a sponge coming up, stalk sponge. Unfortunately, we don't have our sponge expert, Chris Kelly, on the line. So we'll probably just do a flyby of this. Well, Scott is calling this a glass sponge, Ileophus Aerotooth.
All right, wash leads. I would just a quick pilot change. So you want to look at dead center. Is that correct? That's what I've gathered.
Go ahead, pilot.
You want zooms on this, correct? That's what I gathered from the watch change we just did.
Yeah, go ahead, please.
All right, video, come on in.
Should be coming to the end of that ship move, pilots.
Copy that now.
All right, Scott's changed his ID, glass sponge bolosoma.
Is that all you need for the ID, wash lead?
Yeah, that's fine, pilot. We can move.
All right. Video when you're happy. Come wide. All right, so co-pilot, we're moving, what's general direction?
060.
060I.
Yeah, I'm a little off station.
RV nav bridge.
Okay, we moved out of that rather intact lava flow area, which had some pretty interesting volcanic features, big lobes and pillow lavas. Now back into kind of a
move complete. Thanks, Bridge.
debris field, Talus Slope, lots of broken rock of fairly consistent, but many sizes, but larger pieces amongst it.
Watch lead, is there anything in particular that we should be keeping an eye out for as we go upslope? All right, no, that's all right. We can work with that. anything sticks out from your end, just give me a shout, and we'll get good views on it.
All right, all right, Shoreside Science, if you're interested in any specific organisms on the way up, any zooms or anything, just pass that along on the line or on the chat and see what we can do.
Now, when was end of last ship move?
Whiteman Bridge called, so maybe 2 minutes ago.
How long?
2 minutes.
Oh, 2 minutes, copy. Yeah, I guess in a couple minutes then. I think goal was to make some ground.
Looks like I got us ROV distance 300 meters away from the top.
So,
Is this little guy? Video, you want to do a quick zoom on this?
yeah, we have another sea star entering the view just as our biologist is entering the room.
Ready to
So
drive now.
Megan's back. Maybe.
Go ahead, RV now.
Yeah, just confirming bailout due west.
Yeah, you can see the
That's
current
correct.
almost
Thanks,
blowing
Bridge.
out away.
Yeah,
All
so
right.
unlike last expedition, when every time the sea star biologist went to lunch, there was always a sea star. The biologist comes back to lunch, and here is a sea star.
Amazing
What
timing.
luck. So this one looks to be like a Goni Astrid as well, different from the one that we saw earlier. So these are type of cookie star, has much longer legs.
Have we been leaving the ADCP up throughout the whole dive per someone's request, or?
I'm not 100% sure actually.
Okay, I was curious.
Yeah, Sat, do you want
All
any
right.
views on this, or are you happy?
No, we're good.
All right.
I think.
Yeah, but I think it was just to check it every 15 minutes or
Okay.
so.
Yeah, because I noticed it was. Okay.
Dive super.
So that
suggested.
one could have been
Copy.
Ileophus. I'm sure Chris Ma might come in a little bit and let us know what he thinks it is. If he's not on the line, I'm sure we'll see posts about it on Twitter and Facebook later with some identifications.
Yeah.
So was there any interesting biology while I was at lunch?
Oh, we saw some dead sponges and a couple live ones.
Were they stalk sponges?
Stock sponges, glass sponge, I think Bolosomia. That's what Scott ID'd the one as, Bolosoma.
We're definitely seeing a lot more corals in this area.
Yeah, we got into an outcrop with a lot of big lava forms, and it kind of just went up as far as like the primnodes and other organisms all over it. And then we kind of moved off that into a Talus Slope, and now we're back onto more of an intact flank, so it's picking up again.
Looks like we have a Stauropathes right at the bottom of the screen.
Take a couple minutes.
And
you're still kind of.
oh, we did have a
around
request. I
front,
asked
not
what
super
people
far,
wanted to
but-
see. Tina had asked to see a close-up of
Rotate
another
was like 38
Anathemastus.
degrees.
Yeah, okay, I'll call
as
that.
we move up slope.
Sorry, Sam,
Is
did
this
you wanna
one right
zoom?
here good? In the middle of the screen?
Put a driving
That's a nice Anthemastus. This one has a lot more polyps on it
Go ahead,
than the one
Yeah, ready
we
for a
zoomed
move.
into before.
Same
Yeah,
as
we saw
last.
a couple of these on the way up
meters,
that had a lot
bearing
more
0,
polyps
6,
than
0
that,
degrees,
that one that just had
speed
4.
0.2
So.
knots.
Sorry.
Copy that. 30
Science, is that a
meters, bearing
Zoom
060.
request?
Yes, please. Let's zoom on
Copy.
this Anthemastus.
It's a good copy.
That is a beautiful animal.
RV nav bridge, move initiated.
I think we're good for
30
science.
meters, bearing 060, 0.2 knots.
Move initiated. Good copy. Thank you, Bridge.
Tina would like to see a bigger one.
Not quite the size you're looking for.
Yeah, it's not the. If we see a really large one, let's zoom in on it, but I think we're good for this one.
All right, copy that. Video when you're happy, come wide.
So I'm seeing some Narella, an Iridogorgia magnispiralis. Have we zoomed in on one of these Bolosomini?
We did. That was, yeah, shortly before you came in. We got
Okay,
one.
yeah, with the yellow stalk.
Oh, we did see, I guess, a hydrozoan colony on a rock, which was, I guess, kind of unusual, just while you were out.
Well, actually, this one might be a little bigger coming to center, but it's not much bigger. Just want to pass over it.
Yeah, let's pass over it. There seems to be a lot of the same kind in this area.
Copy that.
Yeah, so that sponge is what we've been calling Bolosomini species 1. I think it looks sort of like ET, so in the lab we've dubbed it the ET sponge. Kind of has that double barrel look to the head of the sponge. There are a lot of these Anthemastus, like all over the place.
Yeah.
They really
It's pretty
like your
incredible.
rocks, John.
I guess.
They're really enjoying them.
I guess they are. Should start charging them rent.
Right?
Looks like the ship is about halfway through the move, so you should start feeling it.
It's a lot of
next
small
couple
stuff,
minutes.
though. They're not. I haven't really
That was
seen
a
anything
30
big.
meter move.
Would it be possible to snap zoom on this fan?
The white one?
Yeah, the white fan.
Yeah, there's a video. Come on.
No, Scott had mentioned getting some close-ups of these.
Do you want me to just a snap, or do you want more in-depth views of this one?
I think a snap will be fine. There's a bunch of stuff right here we'd like to see. Tina would like a side of those Anthemastus, like a side view, if possible.
Yeah, copy that.
That way we can see the stalk really well without the polyps getting in the way. Yeah, this looks like another Narella, maybe Narella. I don't want to say dichotoma because the stalk of this coral still has some polyps on it. And dichotoma is known to have fewer polyps on the stalk.
All right, moving on.
Was that good, Science? Sorry.
Yeah, can we look at this black bottle brush?
Is it
brownish,
bottom
right, yeah,
center?
the brownish one.
Yeah.
And then grab a side view of one of these Anthomastus.
So we probably want them set down here, right?
Yeah, sitting down would be probably the best.
All right, video, come on in. Science, what's the name of this one?
This is a black coral. I'm not sure what the name of this one is. There's a number of corals that look like this, and I can make a few guesses.
That's all right. I was just curious.
Yeah, I'm curious too. I have a really hard time identifying these. It could be a heteropathes, perhaps.
Sorry, video.
Or if you nav bridge.
But
ahead, Bridge.
it might be in the Schizopathidae family.
Move complete. Thanks, Bridge.
Tina's saying it's Parantopathes, and that seems right to me. I think I've looked at a picture of that before. So I was just guessing heteropathes because I knew that it kind of has a sort of stalk, but now that she says Parantopathes, that definitely seems like a way better identification. Of course, she is the expert.
All right, is that good enough for you, Science?
That's good for this, and then if we could look at one of those Anthomastus.
Yeah, a sec.
I want to see a nice good side view.
Right here?
Yeah, can we look at that far one on the rock?
Yeah. All right, video, come on in.
And we want to see the stalk.
Copy that.
So this stalk is the same color as the polyps and looks relatively smooth with a few bumps on it. Tina, is there any reason why it's important to look at the stalk of this coral? There's also a corallity, a pink coral, in front of this Anthomastus. It's likely the Hemicorallium abyssale that we saw at the very beginning of this dive. Of course, it might be something
pretty good.
different.
Starting to definitely moving forward now at a good pace.
Copy that.
What is it attached to this rock as well? Or,
Yeah, I think it's attached to the rock as well.
The ship
It's just
stopped maybe
branching
2.5
out
minutes
towards.
ago, so you probably have
okay.
maybe
Got it.
2.5 or 3 minutes left.
Copy that.
Of serious move.
Copy. All right, Science, any more views in this?
I think that's good. Thank you. Tina says those little bumps on the stalk are Siphonazoids. What are Siphonazoids?
Can you size it up, video? Is that good?
Are they a reproductive structure?
All right, compile, push forward.
Copy that.
All right. That was a popular rock.
It was very popular.
That's the social rock of the deep.
You know, it's better to be close together.
Well, this looks like a popular place too.
This is a popular place to be.
You see the current coming down right in front of those.
Yeah, so more of these Anthomastus and Pneumatic, probably Narella. And then I saw a sponge in the background that I'd like to get a look at.
Is it in view still? Or?
It's not in view, it's off to the right.
Oh.
Off to the right.
Yeah, you can see it in Sirius.
Oh,
There's
see
a
two,
number of them.
and then a big dead one. Is that a?
That
That's
does
a big
look
one.
dead.
Big, and two, actually two, or parts of one.
It's a large colony.
That is a huge dead sponge.
One dead center, is that what
So
yes, please.
might have some good stuff up top when we get there.
Yeah, I mean, if Chris Kelly, if you're still listening, what are these dead sponges? They kind of look like that turbocharger shape that we were seeing on that sponge the other day, but it doesn't quite look like it to me. So this is a glass sponge. It might be a Hydroid sponge. Looking at the way that the folds are coming together and how it has these certain holes. These glass sponges have these holes in their body to help push water through the sponge itself.
Right.
And the animals, this is a colony of animals, will feed upon any particulate matter that goes through the sponge. So they're creating, flow through the animal.
So who's causing the water to flow through these
So
holes?
within this colony, there's a bunch of different individuals that have different jobs, and there are certain individuals that help push the water through the sponge by creating a current flow.
Okay, that's an interesting adaptation.
All right, Science, you happy with this?
Yes.
All right, video, when you're happy, come wide.
It might also be a Phareid or Euretid. Thinking maybe Euretid or Hydroids. Some more Parantopathes, Abolosomini. Could we get a zoom on this really dense looking fan off to the upper right?
Yes, you can. Make my way over there.
That looks a bit different than what we've been seeing.
Yeah, I don't remember seeing any fan shape.
This is
more
like
the
a
candelabra
perfect, yeah,
ones.
it's like, yeah, most of them are more candelabra-y, but this one's very dense. Again, I think it's still a primnoid in the coral in the family Primnoidae, but this
Copy
is
that,
a different genus.
Yeah, it should be. Sirius should be settled out right there, I believe.
All right, video, go for zoom.
Copy. Yeah, I'm not really noticing any forward movement.
Yeah, I think 8 minutes ago the ship stopped, so.
Should we put another one in then?
So I'm thinking this might be
Maybe.
Paracliptrophora.
So he's kind of far out, and it'll take a couple minutes to get going.
You can see that the polyps on this
Sean, if you're
coral
comfortable.
are a lot
okay with
smaller.
It. So on Sirius, you can see pretty far.
Yeah, if you have.
And the branching is very close together, and it's branching near the base, and then sub-branching further as it goes up towards the
060
top of the colony.
Copy.
That's where you're facing. Okay. Bridge, RV nav.
Go ahead, RV now.
Yeah, ready for a move. Range 30 meters, bearing 060 degrees, speed decimal 2.
Copy that. 30 meters, bearing 060, speed decimal 2.
Good copy.
Okay, move initiated.
All right, should we move along?
All right.
We should move along.
All right. Video, is there anything else you want from us?
Just trying to count how many branches there are.
So there's been a request to collect one of these Anthomastus, so I'm thinking the best way to get one, since they are so common, is to grab one that is already on a rock. They're pretty soft-bodied, and they're going to be holding pretty hard onto these rocks. So if you see one on a loose rock, we can get a dual sample.
Okay,
I'm not sure if we'll be able to pluck one off with the manipulator arm.
Yeah.
But they're so common around here, so just keep an eye out.
For one on a nice rock, copy that.
Okay, pilots. Ship is starting to move.
Copy that.
All right, another dead sponge stalk. And another live sponge. This is another one of these
Yeah,
bolosomas.
the Bolosomini species 1 is what we're calling that. It has this very distinctive, like, lemon yellow stalk.
Is that the only one that's yellow like that? I think somebody mentioned that earlier in the
It's
chat.
not the only one that's yellow. So
Okay.
there can be other ones that are yellow, but we also don't know, we haven't had an identification to species of that one.
See it?
I think we have some sort of jelly
Oh, yeah.
coming into view. I believe this is it might be a Hydromezusa. Unless it's spherical, in which case I have no idea.
Yeah, if it's too close, no video. Sorry.
Oh, and it's gone. But tomorrow we will be doing some water column transects, so hopefully we'll be able to see more water column
Good chase,
animals.
John. It's coming right at you pretty quickly, I think.
I hear the geology's great on those dives.
Yeah, geology, midwater geology.
Yeah.
I'll take it.
That's good.
It's a new field.
You can be the sole studier of midwater geology.
Yeah, I want to get in on the ground floor.
Oh.
This is quite an impressive
Outcrop,
rock outcrop.
yeah,
Just
this is
a
really
really
nice.
massive feature, really thick flows.
You know why I like geology, John?
Why is that, Megan?
Because when we have really great geology, we usually have really interesting biology.
Wow, that just sounds really good.
Look at all of these corals all over the place. See, the corals, they like really good geology too. So if you're out there thinking you want to be a marine biologist, you're also going to be a little bit of a geologist if you plan on studying any sort of benthic organisms or organisms that like to live on the bottom of the sea floor.
That's right. A lot of the mapping we do, besides just helping with the dive site, is to determine if areas might be good for organisms. Most of the organisms we're looking at here are like the hard bottom substrate, the hard seafloor to attach to. So we typically look for that in the sonar data. We can tell areas that are hard and soft. Soft being kind of the sedimented areas, like you see these sediment
Maybe a local
patches, these
depression?
light-colored patches in between the rocks.
Off to the right.
Yeah, and in fact, I was a explorer in training aboard the Okeanos Explorer last year and learned a lot about mapping, which has really informed me about these operations that we do with the ROV and helped me in my research and understanding where we might find coral communities.
Let's see, 25. So 6 minutes ago, I called the move in.
That's
so
right. This is your third
you should
trip
actually
out
just-
here, right?
Yep, this is my third time.
What was the beginning of your move?
I really love this ship.
Should start
We
moving
have a
soon,
great
30
crew
meters.
aboard. Oh, there's some really interesting sponges over here. And some
Copy
pretty
that
cool
bridge.
corals.
Move's complete.
This looks like a,
I think I
looks
see.
like a chalice.
Yeah, so this looks like polyopogon
the
to
ship
me.
is done moving, so
It's a
probably
type
5, 6
of
minutes
sponge
ship
called
move coming
polyopogon
your
in
way.
the family I can't even say it now. Pneumatidae. Pneumatidae. There we go. Some of these names, they get so long.
I'm glad rocks don't have Latin names.
Well, some rocks have some challenging names too. I can't think of any at the moment.
That's nice.
They like to end in like "ine," like olivine and.
Oh, yes, yes. Those are minerals of significant
I
interest.
think that's a good zoom on that. There's another sponge a little bit further down I'd like to see as well.
Further down, copy that.
So there's a
Video when you're
benthic
happy
on the top
tomorrow.
of this sponge. Yeah, that little translucent one.
Is it still
That's
down?
right in the middle.
Oh.
I'm not really sure what this sponge is. It looks like a glass sponge, though. I'm trying to determine if it is three-dimensional or more of a leafy-looking sponge. Yeah, so this one looks like a leaf. So that's very interesting. It's different than the other sponges we saw today. So it could be a euretid. They tend to be leafy in some locations. Conolasma might be a good identification of this. Too bad Chris isn't online to tell us a little bit more about what this might be. Some of the characters we're looking for include the different surface texture, and of course to identify these sponges we need to look at the spicules. So spicules are the little glass pieces inside the glass sponge that make up the matrix of the skeleton. And every different species of sponge has a very unique set of spicules.
All right, can we?
We're good for science.
All right, video. When you're
So
happy.
is that growing into the rock there? Or is there a base that's attached?
Yeah,
like
the base is
horizontally?
probably attached to the rock. And it probably chose this location because there's a current coming
happy
through,
video, come on
and it's
live.
allowing the water to pass through the fan of the sponge.
Oh, no, that's right.
Do we know how these sponges propagate? How do they find a new location? I know corals have larvae that are expelled and settle. But what about sponges?
Sponges also have larvae.
Okay.
And they disperse in a very similar way to corals. And corals do have different strategies of reproducing. Some are spawners, which means they expel their reproductive sperm or oocytes into the water column, and they have to come together, and that new coral zoid will, or what's the term? The new coral
Trying to help you.
Yeah. Why am I asking the geologist for the biology term? I can't remember.
The candelabra.
Is this rock loose right here at the bottom of the screen, you think?
With the
Anthomastus on it?
Yeah, it would be a
right there. There's
rock.
one with two on it.
I was going to iron that one too. Do you want me to give it a poke?
Yes, please.
Got that. Nav and Copilot, we're in a good spot, right?
I think so.
What was this?
We'll let you know if
Oh,
not.
the coral
larva. Yeah. So the new coral larva then
it's 36,
swims
so
about in the
minutes.
water column and then settles
so if you have
down
any move
onto
left,
a place
it's
to
not very
live.
much.
Okay.
So
Otherwise,
we'll keep
corals
an eye
can
on
be brooders, which means they have their larvae start their lives inside the tissue, and then once it gets to be mature enough, it is then expelled to go find a new place to live.
Thank you. Can't quite get my tongue there.
Can try to fit in a bio box then if you get.
Yeah.
Yeah. Obviously.
Alright.
That rock does look loose, though, and you get 2 for the price of 1.
Yeah, this would be great if it's this. Alright, arms come live.
Bonus samples.
Bonus. It's always good to have a secondary sample of these guys, because if one gets damaged, you can also keep one intact, and one you can open up and look at the inside of this coral. So should we count this as a biologic or geologic sample, John?
I think it should be counted as a biologic.
Okay. You need another rock?
It's loose.
It's loose. All right.
I think you'll
Let's grab it.
Yeah, I think you'll want this in your bio box, right?
Yeah, we're going to need it in the bio box.
So you don't.
Of course, I don't think the Anthomastus will bail on us.
Okay.
But I think they'll be a little more protected, and when they come up to the surface, they'll be in water. So in our geo boxes, there are holes that let water out, but in our biology boxes, the animals stay in their nice cold water and will stay fresher longer after we come to the surface.
Tilt up for you, pilot?
I can get it.
Okay, sorry.
Oh, actually, yeah, if you want to do it then. Yeah, go for it, and then I'll rotate for it. Alright, Biddy, if you want to get zooms on this.
This is specimen 3.
I believe so.
Do we have any, or are either of the port bio boxes available?
Port inner, I believe.
Port inner.
Alright.
Port. I got port outer.
Port outers. Open or
is occupied.
Yeah, so port
Okay.
enter should be good.
Yeah.
I think it'll do perfect.
Yeah.
Last pilot said it wasn't a floater, so yeah, port enter.
Copy.
That works. Can you open it from here?
Yep, I think so.
Great.
You ready?
Yeah, go for it.
Okay. Coming out. Oh, looks like I'm pushing you.
Yes.
All right, I'm coming back in.
That's all right. I can back up real quick. One second, watch these. I just have to back up to get the drawer open. Before I can do this.
I'm going to put the mini Zeus in the port biofilm.
Copy that. Alright, that should be
All right, try and come
better.
out again. Coming out slow.
Nice. That's great, thank you.
Looks like it'll fit nicely in there.
Beautiful.
Alright, go for
Yeah, and those corals don't look too worried.
Closing.
They haven't even pulled their tentacles
sample
all
shot.
the way in. So they'll be nice and safe and sound until they get to
Okay,
the
we're stowed.
surface.
Nice, thank you.
All right. Hopefully we'll have some good stuff to look at and preserve.
And a bonus rock just for you, John.
All right.
Alright, arms off. Great.
Now these Anthomastus corals are just really
your,
prolific in this
just off the
area,
Cirrus
so
port
having this
side there.
collection is
Yeah.
important
Oh yeah, if you want to take your camera
so that
back.
we can really understand what species this is
set up,
and maybe get a sense
Okay,
of
I kind of
their population.
swung a little bit to port to try and keep
Oh
you
my
at
goodness,
least
that
light
rock
pool
has a
view.
ton of them. When we
Wow.
annotate this video, it's going to be hard to count them all.
Have uphill
1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11,
Yeah, we
12.
might have to
Yeah, this is the point where I go,
a little bit to port.
looks
maybe.
like about
Yeah.
30.
Yeah.
So that's about as far as I can go that way.
Yeah, it looks like the local high is almost due north.
Yeah, so then upslope right now is
From Sirius to global high, I got 070. So maybe we go kind of northbound and then turn right, probably. So yeah.
This does
You
seem to
got-
be a more spongy dive than yesterday.
that's
Spongy.
Spongy.
Say again, Pop?
Keep seeing a lot of really interesting
instruction.
sponges.
Yes, with that.
There's this
Yeah.
big white sponge.
Yeah, I saw that earlier,
40.
one like it.
040.
Yeah.
But I was wondering about
you
what was
comfortable
going
with
on with
Yep.
that.
Co-pilot's comfortable.
Good.
And the thing with those is,
I'll do 30 meters. 040.
Okay, I'm at 040
well,
now,
I
pilot.
know a little bit about
Copy.
them because
Bridge,
Chris
Harvey, now.
has been teaching me a bit about these sponges.
Yeah, I would like a move of 30
this one looks pretty
meters,
interesting.
bearing 040
zoom on this
degrees,
side?
speed 0.2 knots.
Yeah, I believe so. Let's let's zoom in.
Definitely a glass sponge. It could be this Hydroids that we saw, I think earlier, or it could be something else. This is really interesting. What are some other? It could be a Phreid or a Euretid.
It's not the lettuce sponge,
Do
is
they
it?
do they look like fully formed tubes to you, or more lettucey?
I thought lettucey, more like. Well, there's some here that
that look
look more tubey, but most of them I thought looked.
Look lettucey.
Yeah.
So the lettucey-ness of it makes me think that it is a Euretid. And it could be, we don't really, so like, check this picture out on our animal guide. It kind of looks like that. And we just have that family Euretidae. It's very interesting, that's for sure. It could also, well, it doesn't really look vase-like, so it's probably not the Periphergella that we saw, I think, a little bit earlier and yesterday.
All right, there was Megan McCullough mentioned that there's a urchin to the left of the sponge. I did
don't
not
think
see
we've
it.
seen any urchins yet today.
Pilot, if we could maybe take a look, zoom out, and see if we can see an urchin.
I don't think I'm seeing any. Are you seeing any?
I'm not seeing a urchin.
Yeah, me either.
Maybe he was a high-speed urchin and he scooted away.
Yeah,
all right, I guess, is there any reason to see the inside of this, or we can move along?
We can move along.
Okay.
All right. Pilot, I'm going to recenter a little bit with you.
Copy that. I was getting pulled off a little bit.
Oh, she says like right near the sponge. Well, I think we're moving away. I suppose maybe it was pretty small that we didn't see it.
Okay, I'm rotating.
All right, we had a second urchin sighting.
So the fact that we see all of these Anthomastus suggests that there's a lot of local recruitment. Scott was saying that perhaps these Anthomastus corals have crawl-away larvae, and they settle very quickly away from their parents. So these might all be related to one another, and that also implies that they have internal fertilization.
That sounds rather antisocial.
Well, they just want to be close to home, you know?
Yeah, okay.
Nice, big, happy family.
So that's easy. It must just be a local impression.
Local?
Yeah.
Yeah, we'll keep going up, see what we see.
So we're getting into a little bit more dramatic topography, more bumps and hills and valleys and little small chasms. It's kind of more interesting in that way, but still kind of a massive lava outcrops. Not a lot of detailed features to see. It's all covered by this manganese crust, so that doesn't help things.
Cereal should start to move here.
But at least it's given us something to climb over and something for the organisms to attach to.
And we've got a lot of fuzzy stuff along on these rocks.
Well, we saw that earlier while you were at lunch, but not to this extent.
Yeah, is that the hydrozoans?
Yeah, in that case, though, they were kind of isolated to one single boulder of medium size. And they're like all over these outcrops, these peaks.
Well, that means that this must be a really great place for biology. There's just every surface is covered with something. But we're also seeing quite a bit of diversity with anthomastus, primnoids, hydrozoans, sponges.
So Les has some interesting thoughts on the reproduction. I don't know if...
Oh yeah, so looking in the chat log, Les is saying that if you see several large, massive colonies, these colonies all of the same size, that would suggest that there was a massive larval settling event where they're all recruiting at the same time. And we've been seeing a lot of different size groups, some large, some small. And that would suggest local reproduction and recruitment, or asexual reproduction. Tina was saying that these anthomastus might, what was that? They lacerate, and then they must move away from their parent colony. Oh, there's a teeny tiny Goni Astrid. Let's try to get a nice zoom on that guy. Or maybe it's a different type of star. It's pink, though.
Yeah, notice that.
So I think these hydrozoans are similar to the ones we found stuck onto your rock that we collected yesterday.
Oh, okay. I had to pick those off carefully with the tweezers.
Mm-hmm. They're very fine and very small, but these ones are very densely packed and a lot larger. Well, it is quite adorable. And it is definitely a Goni Astrid sea star. And I would love to collect it for Chris Ma, but I really don't think we'd be able to get something that small on this rock.
Yeah, I think it's centimeters.
Yeah, that would be just too hard.
Yeah.
But we'll get some good imagery, and hopefully that'll help Chris out in the future.
I think I saw some of these at the souvenir shop before we left.
Right?
Yeah. We'll just get him a ceramic one and see if he can ID it.
Idea.
Video whenever you're happy with that.
Yeah, we're happy with that.
Great.
Thank you. Now, if that little guy was on a rock or something, maybe we could have grabbed it, but the cookie stars from these depths are not very well known. And so Chris Ma at the Smithsonian's been working on identifying these and learning more about them.
Did he ever say what got him interested in sea stars? Or do I need to ask him that next time he calls in?
I think you should ask him.
Okay. Sounds like you've heard the story.
Yeah, well, you know, he did do some schooling in geology, and then just, he's a convert, you know.
Yeah, well, those
came to the squishy side.
The squishy side.
That's probably his first experience with the slime star.
He began on the crusty side.
Oh my goodness. Look at this forest of anthomastus. They're just everywhere around here.
Look at these outcrops.
My biology is obscuring your geology.
It is. At least it looks like we're progressively getting some larger sea fans, these primnoids or candelabras, and more of them as we're moving up.
Yeah, so I'm thinking a lot of these are Paracalyptrophora. Some might be Norella. I'm seeing more of those thin leaf-like glass sponges.
Okay, so depth readings.
There seems to be either a really large rock in the background or
So
a really nice
depth versus
sponge or coral. You see it?
maps. So you're at 238.
Yeah, in the back, the back upper left.
The back upper left has some white.
That's about right.
Looking.
Something large.
Large.
A lot of dead stalks here too, of sponges.
All right, you want to get another move in?
Yeah, if you're ready.
And a big yellow one.
Ready for another move?
Oh, that's a really big Bolosomini.
Direction 040 still look good.
Species one.
What do we do? What do we look left or right?
Well, we've seen that.
Looks like a local high
that
for
Bolosomini
Sirios.
before, the yellow one. So let's look at the big white one.
Copy big white.
On the left, sorry.
Might be just out of reach, but I might be able to get us pretty close. It's been like 5-0 or something. I think I might turn back around.
050.
Yeah, I think it'll be fine.
Bridge RV nav.
Go ahead, RV nav.
Yeah, I'd like a move of
talk to
30 meters, bearing 050, speed 0.2 knots.
Copy that. 30 meters, 050 degrees at 0.2.
Oh, there's a yellow coral on this rock too.
All right, video. When do you come
I'm
on
loving
in?
the spot. Now this is different. So look at the way the spicules are lined up, and it looks like striated almost, like crisscrossing pattern across the sponge.
RV nav bridge, move initiated, 30 meters, bearing 050 at 0.2 knots.
So this
Move
looks
initiated.
like
Thanks,
a
Rich.
soft sponge instead of a crunchy, hard sponge. Thinking Corbitellini? Yeah, I'm telling you.
Interruption. Your conference contains less than 3 participants at this time. If you would like to continue, press *1 now, or the conference will be terminated.
It sort of reminds me of that
Copilot, I called that move
giant
in at
sponge
050.
that we
so
saw
just a little
in the
change
Northwest
that right
Hawaiian
there.
Islands.
Just thought I'd let you know.
Copy. Thank you.
Yeah.
But it's not quite right.
All right, pilot. I'm going to swing around to 050 then.
Copy that.
That'll
Yeah,
get
let's
you
move
right
on.
the edge.
I'm going to look at my pictures real quick.
And Copilot, I just called the move in, so you got about 5 minutes until you start moving.
Science, are you happy with those views?
Yep.
Great.
Yeah, so the best I can go with that is probably Corbitellini. Perhaps a new genus. And so Corbitellini is the family Euplectellidae, and that is a glass sponge. Now this one just seems not very consolidated, and usually these things are more of a vase shape, but this one is quite large and just flopped over. Very interesting.
Yeah, it's unusual.
Chris Kelly will be excited to see these sponges. Now that's a really nice looking Bolosomini. Look how yellow it is.
Yeah, he's yellow all the way to the top.
So again, this is what we're calling Bolosomini species 1.
Or she. Do these sponges have genders?
Well, they might actually be both.
Oh,
I'm not sure.
how convenient.
How convenient.
Sirios, the brightest star in the sky.
What about corals?
So some corals can be both, and some corals do have genders.
Interesting.
So Sean, from you to waypoint 2, the top of the hill, I got 77 degrees. So that's about right.
77. 7, so I'm looking, yeah. Should be pretty good.
Copy. Maybe the next move will adjust our heading a little bit.
Yeah.
I think.
Me and Sirius are almost back in line.
Yeah. Sounds good.
And there's a little baby one. Little.
Was that?
Watch it.
Oh, he did. I couldn't hear. Must be turned down. Oh yeah. Here we go. Go ahead, watch lead.
Yeah, I was just wondering how
Yes. Looks, according to my estimations, we'll make it to the top. No problem. We're right on schedule. Yep.
Okay, we had some feedback on our chatter. From Tina says black corals are boys and girls.
And the tube anemones that we collected earlier on that sponge stock are generally both.
Okay. That's what the experts say.
Yep. Now, if you're both, what would be interesting is, are you self-fertilizing, or do you have to find a partner in order to reproduce?
I do not know the answer to this question.
That's my question.
Well, now, as you've probably noticed, we've moved off of these big outcrops with a lot of relief to another kind of a consistent bottom on a slope with a lot of little small rocks, small cobbles, and some a little bit larger than others. Still a fair number of these, what are the? Anthomastus. I keep wanting to say something similar.
But yeah, it's not as dense as when we were in those giant outcrops.
Yeah, now see, it looks like they preferentially pick the larger cobbles, at least the ones that have survived, right? Unless these are crawling ones, as
Go ahead, Bridge.
Scott had mentioned.
Yeah, I'm not sure if they can move. Yep. Part of me thinks that they should be able to move.
Yeah.
But yeah, if you are one of these corals and you're on a small cobble and a nice big current comes through, you might topple over. And if you're not a fast mover or you can't move, that won't be good for you. You want your tentacles up into the water column to collect food. Down at these depths, there isn't a lot of food availability, so you need to catch what you can.
Right.
And Scott is saying that larvae that settle on smaller cobbles may not survive as they grow.
Right. And thus, we're just left with the ones that landed on the big ones, or got to the big cobbles or big rocks. So.
It's the circle of life.
Yeah. So it's the three pigs analogy, right?
Mm-hmm.
Don't live in a straw house.
Something like that.
Copilot Nav.
Go ahead, Nav.
We used to have black tape over the touch buttons on that monitor. I think it fell off. So
oh wow,
I
look at the size of this
the other day.
sponge.
Oh, over in
the...
Okay.
So be careful of that.
That is good and huge. Like, look at the lasers. They're 10 centimeters apart. You can almost barely see them.
Yeah, I remember that during the
This
move.
thing is a couple meters long.
Calibrate it.
Yeah.
It looks like part of it's dead, but there's still a lot of alive bits.
Swinging around. I think you're still in pretty good position. Off station.
It's quite the colony here.
It is.
Yeah, I'm starting to
Can we
catch up to
zoom
you.
science, or you just want to kind of pass over
Let's
it?
zoom on this. I want to see if it has the same sort of structure.
Copy that. Video, go for zoom.
That's interesting.
So is
It
this
looks like it has an internal structure.
this is different than anything we've seen this dive, or similar to the other one? One of the other
I think
ones.
it might be similar to the Corbitellini that we just looked at, but at the same time, I don't know. This looks a little strange to me. A little bit like the Languinility ruffles that we saw. I think that was dubbed the minivan sponge. It was almost as large as the ROV.
Was this the one, the largest sponge ever?
It looks a little bit like that to me, but that sponge didn't have this surface patterning in the same way, which makes me think it's still Corbitellini. I think we're good for science on this zoom.
All right, video. Anything else you want with that? If not, pull up. Great.
Very
Size it
interesting.
up if you want.
I'll be interested to see what Chris Kelly thinks about this sponge.
Starting to catch up with you,
Best
Pilot.
sponge ever.
Yeah, I'm not sure
in
if
good
it
position
fell
right
over or it started growing like that. But there's definitely this back end is in the process of
Looks like that's
dying,
living,
and
and that's
and this is and
yeah.
and it looks like it's getting colonized by other sponges.
I think he's confused.
It's a very confused sponge.
Okay, I think you're done moving. You guys ready for another move, or maybe change the heading a little bit on this one? Or?
Let me center and let's see where we want to go.
Yeah, it seems like local high is a bit, closer to what you were saying, the 7-0.
Okay. Yeah, so from Sirius to the top on high pack, I got 75075.
Yeah.
But that's global.
I'm looking 7-0 right now.
Okay.
That looks pretty upslope, and I'm in line with Sirius.
So maybe 060 for Sirius? Or do you want to do?
Yeah, we can split it, right?
Okay.
That'd be good.
Okay, I'm pretty much already there.
So if you're just joining us, this is Megan Putz.
And John Smith.
We're from the
060
University of
on Sirius
Hawaii,
pilot,
and
so
we're
we
here
lined up.
coming to you live on the
Sirius
Okeanos
2.
Explorer
Yeah.
at Beethoven Ridge.
Pretty
We're up
driving,
in
yeah?
the
Great.
Musician Seamounts, which is located
Yeah, we'd like
north
to
of the
move,
Hawaiian Islands.
30 meters, bearing 060 degrees, speed decimal 2.
We're moving north as we go. This is our third dive of the cruise. We left on Wednesday,
Copy.
and we're heading north and eventually to the west and looping around after we do 20-some dives.
Right now we're on a small cone feature at the top of the Beethoven Ridge, and we're transecting up to the top of the cone to see what's at the very summit of this ridge. So
Good copy,
far,
Bridge.
we've seen a really dense community of Anthomastus corals, Primnoid corals, sponges. It's been very exciting.
And we've seen a variety of geological landforms, like what we're seeing now, Talus Slope, which is a bunch of broken rock of various sizes, sediment in between. Seen some pretty dramatic lava flow outcrops, pillow lavas and lobate lavas, and just some big massive walls.
tomorrow we will be diving on Sibelius Seamount, and after our bottom dive we'll be doing some midwater transects looking at the midwater animals. The midwater, which is makes up pretty much most of our world.
The largest biome of the planet, right?
Largest biome on the planet, has been very little explored. So having this opportunity to transect through the water column and take a look at the animals that live there is a great opportunity to learn more about them.
It'll be a jelly-rific dive.
Sure will. John's going to become the first midwater geologist.
That's my goal.
Pilot, can we zoom on this long coral right in our foreground, on the right side?
Yes, we can.
I want to see if this coral is a bamboo coral or another unstalked or single stalk primnoid.
All right, video, go for zoom.
Yeah, so this is a primnoid coral, and I think this might be Candidella gigantea. To tell this coral from the other primnoids we've been seeing, there are these little scales along the body. Candidella is known to have many small body scales, sort of overlapping, looking like fish scales or dragon scales. And you can definitely see that around the body. Right here we have a whorl of 5 polyps. So every time there's a grouping of polyps, it looks like there's about 5 that are going around the outside of the center stalk. Very beautiful.
Why is it gigantea?
Because the polyps are quite large.
Good answer. Are these spiny things inside the? Are these little?
So those
spines?
are part of the
That's
body scales.
Okay, those are the scales.
Tini is saying that sometimes you can get up to 6 polyps in a whorl.
Maybe even 8.
That's a very crowded whorl.
Are you happy with that science?
Yes, we're happy with that. Thank you.
All right.
The best of both worlds.
I like it. So that was definitely a new species that we've seen today. And new for this expedition so far. We have seen Candidella gigantea in other locations, in Northwest Hawaiian Islands, and I think also in the Marianas, possibly even Wake Island.
You said they also look like dragon scales?
Well, fish scales or dragon scales.
Which dragons have you seen?
I haven't seen any dragons. Well, except if you watch Game of Thrones.
Oh, okay. Just checking.
I just like dragons.
There are Komodo dragons.
Komodo dragons.
Yeah, but I don't know if they have
lizard
scales.
scales.
Yeah.
They do have scales.
They have scales.
Yeah.
They look really smooth.
Yeah. Well, they look smooth, but they are plated and overlapping. Snakes have scales that way.
That's true. That is true. But we don't have snakes in Hawaii, so I haven't seen a snake for a while.
Yeah. I'm very appreciative of not having snakes in Hawaii.
Yes.
You know, you can go walking in the woods and you see something that looks squiggly on the ground and you know for a fact that it's not a snake.
No poison ivy either.
Also thankful for that.
These are our public service announcements for the Hawaii Tourism Authority.
Yeah, upsides of traveling to Hawaii. Not only is it beautiful, but no snakes and no poison ivy.
No bears.
No bears, no squirrels.
No lions. No tigers.
Oh my. Tina says, but we still have spiders. Yes, we do have spiders, but we don't have any venomous ones. At least not that I know of. Do we have venomous spiders?
Nothing deadly.
Yeah, not like black widows or anything.
We have the cane spiders. That's in. I probably imagine they could give you quite a bite if it might bother you a bit, but I don't think they're poisonous to humans.
I think the worst thing that I've seen in Hawaii are those really giant centipedes. I don't like those.
Apparently Scott France likes snakes.
I like snakes too. I just don't want them to bite me.
He says they eat the rodents. I guess he's not a cat fan. Maybe.
Yeah, cats eat rodents. Or mainly they just bring them to you.
Hey, look at this line of these. Anthomascus. It almost looks like they're in a line going up the ridge.
Yeah, they're marching along.
Yeah.
Maybe like they reproduced and like split off, or they can reproduce asexually and just moved a little bit and they've progressed along this ridge. They're all lined up perfectly.
Next pitch.
They have marching orders.
They look like, actually, they look like these windmill farms we have in Hawaii and that are popular across many other areas now. But they go up the ridge line on the islands like Maui and Oahu, and it's like little windmills. Wind turbines, I think is the right description.
I got a good view of those when we were headed north, looking down at the North Shore.
Oh yeah, on the cruise.
On the cruise. It was quite cool to see them from the ocean side. This is quite the dense community here. Seeing a bunch of Anthomastus, some Primnoids, some Bolosomini species 1, some glass sponges, which I think are
guys look like you're ready for that.
Conolasma.
Co-pilot's ready.
Yeah, I'm stretched out nicely.
Some Corbitellini.
060 looks pretty good actually.
Sounds good. Yeah.
Yeah, I'm fine with that.
Okay. Bridge RVNav.
Go ahead, RVNav.
Yeah, I'd like to move, same as last. Range 30 meters, bearing 060 degrees, speed 0.2 knots.
Copy that. 30 meters. Bearing
Big Yaller off to the right.
Yeah.
Good copy.
So much sponginess.
Move initiated. Thanks, Bridge.
Yep.
Now these Bolosomini, they're actually quite soft. So once we had one that we had tried out, and it was very soft and, well, as you would expect, spongy. You know, and some of these glass sponges are quite crunchy. They have a thicker density of their spicules. They can be quite hard.
Kind of like a, I don't know, cotton candy past its due.
Mm-hmm.
Or like fiberglass, something like that.
Well, we've got a water column animal.
Video, you want to go for this?
This little water column. It's a jelly. That's my identification.
Wow. So that all that we're seeing is the one organism, right? It's
Yeah, it's all
wow.
one organism, and I think that long line was its feeding tentacles. It was really cool.
I have
No
never seen one like
idea
that.
what that is. I don't know my jellies.
We'll find out tomorrow.
Tomorrow. By the end of this cruise, John, we'll both be water column initiates.
Initiates?
Yeah. Initiates into the world of water column.
Can we be water column associates?
Associates. Oh, check out this like broken pillow right here, John.
Yeah, that's kind of a classic example. You can see the whole intact sides, the cylindrical part. And then on the face of it, I call that a fractured pillow. So that basically just broke off for some reason, whether it just weakened over time or got shook up or got hit with another rock or something. But we see that fracture pattern where you see those kind of spoke-like radial grooves. Those are kind of where the prismatic talus falls out, so kind of triangular shaped pieces come out of that and slide down slope and make all this talus or broken rock that you've seen us traversing across.
Tara suggests that we should be water columnists. I like it.
Water columnists.
We're water columnists.
That's only if you publish on the data from the water column.
Yeah, then you'd well, we are going to be writing our dive report.
Okay.
Would that be a columnist enough?
I don't know, it might be a creature feature.
Creature feature.
We started moving maybe 4 minutes ago, so Sirius should start moving in the next minute or so.
Copy that.
The dense community continues.
Yes, and more fractured pillows. So the slope looks like it's picking up a little, steepening, and we're seeing a few more of these fractured pillow faces.
Scott's suggesting that we call this seamount Mount Mushroom. For all the mushroom corals, these anthomastus corals that we're seeing all over the place. This is the cone of mushrooms, mushroom cone.
Cone of mushrooms.
ROV nav bridge, move complete.
Move complete. Thanks, Bridge.
The sponges are picking up.
Yeah, I've noticed quite a few.
Quite a
Moving
few.
up.
I keep seeing them all over the place. So I've seen a number of these Corbitellinae colonies that we've been calling Corbitellinae new genus. They're these space-like colonies that have odd projections on them. We have quite of a large coral up into the upper left, if we could get a look at that. Now this actually looks like it might be a rosellid. More
This
like,
sponge here?
yeah, this sponge right here looks like it might be that Languinellity. 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 Trying to figure out how to pronounce it.
Science, you want zoom on this?
Yes, please.
All right. Video, go for zoom.
Oh, it has a sieve plate on top, but it is quite fuzzy.
What do you call that on top?
A sieve plate.
Sieve plate.
So it's sort of covering the opening of the vase. Or do you think it looks more open, actually?
It looks open.
It looks
to me.
open.
The pukas, or the holes, are look to be in the sides on both that side and the foreground.
Yeah, I see that. Yeah, so it isn't closed. I was gonna say, I'm like, oh, these are supposed to be open. So, yeah. For those of you who aren't familiar with the Hawaiian term puka, that means hole.
Looks like it does need it's got a little 5 o'clock shadow around the edge there. It needs a shave.
There's some hydrozoans up top, and then it has
You guys
these
are ready for me
dense
to call in another move?
areas of
still looks good to me.
spicules sticking out from the sides, making it
What do
look
you guys
very
think?
fuzzy.
Yeah, sounds good. Sounds right.
All right. Do we are we done with this one?
Yeah,
Megan?
I think we're good
All
with this
happy?
one.
Great. Video, if you're happy, come wide. We'll keep exploring.
Bridge RV now.
Oh, that's interesting down there.
Oh yeah, can we look at this
like
little other
Sinus
vase
last
next to it?
30 meters, bearing
Yeah,
060
right to the left.
degrees, speed 0.2 knots.
All right, video, go for zoom.
Yeah, so this is
Move
looks like what
initiated
we've been
at
calling
30
Corbitellinae new genus. It's a Euplectellid, so
initiated.
it's in the
Good copy.
family
Thanks, Bridge.
Euplectellidae. And the sponge we just looked at it, at that's right next to this one is in the family Rosellidae, and they are both glass sponges in different families, and this is in the subfamily Corbitellinae. And it has these really interesting projections on the side of the colony.
Yeah, that's pretty crazy.
We're good for science.
All right. Video if you're happy. You know the drill.
So very exciting. A lot of different kinds of sponges all in one place. Okay.
So now, what structure move? Still
060.
Copy that.
So it looks like we have a Bathopathes where the top half is dyed out, and there might be some barnacles and maybe a crustacean and other crustaceans along the stalk that's empty now.
Yeah, video go for zoom.
Ah, yes, they are barnacles. So these are stalked barnacles that are connected to this Bathopathes skeleton. And then lower down, I think I saw a crab or a squat lobster.
Down. Yep, right there.
Oh, yeah, this is a squat lobster. I think we were calling this one Galathioides, spiny.
Galathioides.
That's the best identification we've had of this guy. Mainly because every time we've tried to collect a squat lobster like this, it swims away. They know what's coming.
All right, science, anything else in here?
I think that's good. We're
All right.
good to go.
Video, if you're happy, come on out.
Co-pilot, I called the move in maybe 4 minutes ago, so you should start swinging in a minute or so.
Copy that. Thank you, Nav.
Yep.
So again, giant fields of Anthomastus. Now we're seeing a lot of different kinds of sponges. Oh, I think this is a Relicanthus.
Yeah, making my way there.
Our data manager, Nolan, said that this is one of his favorites.
All right, video go for zoom.
So while this might actually look like an anemone, it's in its own group.
Summary.
It's in the family Relicanthidae, and it's not as related to an anemone as you would think. It's really interesting that the tentacles on this Relicanthus are
Go ahead, Bridge.
spirally looking. I
Moves
haven't
complete. Thanks,
seen
Bridge.
that look like that before.
Yeah, it's a nice one.
Maybe, Nolan, could you tell us a little bit more about Relicanthus?
Yeah, pretty long. Copilot, you want to get lasers off so you can get a nice view without them real quick.
Roger that. Oh. There
Nice.
you are.
Thank you.
Rob.
And Nolan thinks that the spiral tentacles might be retracting to the oral disc, or that's the center of this animal where the mouth is.
All right, science, are you happy with that?
Yep.
Great.
That's pretty amazing.
Video,
It's
you got
pretty
what
cool.
you need. Let's come wide.
I really like
Then
them. The long
Copilot,
tentacles
please
are
get
just
lasers back on.
super neat.
Roger that.
Nice.
I'm going to look down a little at you. We're getting a little more overhead now.
Yeah, copy that. I'm going to push a little forward.
This is quickly becoming a sponge garden. In the contest of sponge versus coral, I'm not sure anymore who's winning.
That's right. We didn't have enough participation early on to have that wager. But, I'll carry it through from yesterday.
It seems
good,
like
pilot.
as we get
Got a
close to the
45
top
degree
of this
angle on you right
feature,
now.
Nice.
we're seeing more and more different kinds of sponges. Yes, those
We saw that one.
we saw that tall sponge before. That's
Right.
Poliopogon.
The one that looks like a chalice.
And then that round one that was near the bottom, that was also a Poliopogon.
Do you want any zooms on anything?
Can we look at this sort of fuzzy colony in the bottom right?
Bottom right.
I think this might be a Chrysogorgia.
Right.
Coming around right where the lasers
That
are.
one?
Yeah, I think so.
Yep,
All right,
that's the
video,
one.
Come on in.
Yes, I think it is a Chrysogorgia. Now these Chrysogorgia corals usually always have associates. Yep, and I see a little squat lobster hanging out inside the branches.
Is it in video?
That squat lobster is probably Europtychus.
Tina's asking if it's Paragorgia or Scots. Or
It'll rotate briefly.
Oh, next to the Chrysogorgia, there's an Iridogorgia.
I'll come back.
So those are both Chrysogorgia corals in the same family.
All right, science, are you happy with that?
Yep.
All right, video, come wide. Yeah, and Copilot, you can just, as I come back, you can kind of
copy that.
Readjust.
Oh, it's getting hard to see the rocks.
I know.
About 10 degrees right now. I'll follow you back to 60.
Awesome.
Oh, can we
sorry,
look
we're,
at
we got a
this
heading
pink
30 right now.
coral on
Okay,
the left
copy
side?
Oh, sorry.
Sort of pinkish red.
Pinkish red.
It's now coming into the center screen.
It's up a little bit, I think.
Up a lit from the
Side-
lasers.
yeah,
You're
we're
kind of looking at the
catching
profile
at it
of it.
sideways.
It's now on the right side of the screen.
It's right by your
Oh,
right
there's another
Oh,
one.
there's
down.
another one.
So
Right now in the middle.
Is it dead center?
yeah, I can go for
I believe that and the
Alright.
one that we just saw are the same.
Video, go for zoom.
even I would agree.
If the geologist agrees,
Is this the one you want or the one to the left?
the one
Like a
further
bubble
up,
wrap
the
coral,
pink
possibly.
one.
It's just to the left if you
Left,
zoom.
yeah.
Is it? Right there. There it is.
There we go.
There we go. Okay, yeah.
Ah, yes. It is a bubblegum coral.
Hard.
Bubblegum. Why is it bubblegum?
Because doesn't it look like bubblegum? It's a very pink. So this is a Paragorgia.
Okay, I think you guys are ready for a move.
And then on top of this
wait,
coral,
wait,
we have an Astroschematidae
Yeah, let's let
sea
Pilot get
star,
streamed out in
or
front of
brittle,
us.
or not basket star.
That's all right.
Boy, that's tough to focus.
Yeah, sorry, video. There's way too many things on the ground to set.
Don't want to sit on anything.
Yeah.
Scott says
All right.
it looks, when the polyps retract, the tips of each of the branches look like a bubble gum ball.
Ah, okay. All right. Are we good on this one?
I think we're
this
good
zoo?
on this one.
Move on up.
So this is definitely the first time we've seen a Paragorgia today.
Video. Awesome.
So it's good to get nice video of that.
Well, this dive started out pretty sparse. And like a lot of them,
Looking good.
getting
Pilot.
better
I'll
as we
follow you
move
you
to the
come
top.
around.
Nice.
And I was a little worried at the beginning of the dive that we wouldn't have a dense community, but I am pleasantly surprised.
You gonna come back tomorrow?
I think I will. I think I will come back tomorrow.
It's good to have choices.
You know, I might just come back every day for the next 20 days.
Wow.
That's how much I
That
like
is
it.
All right,
commitment.
that's looking a lot better already. I have a heading of about
You could be doing so many other things.
I
try
could
60
be, but this is
degrees.
what I would
We're trying to
choose to
catch
be
up here.
doing.
Nice.
This is definitely my dream job right here, exploring the bottom of the
yeah.
ocean,
Think you can get a move in then?
seeing things that no one has seen,
060 still good to you both.
So I'm looking 6-0, and that looks pretty
and
good to me.
exploring with
That's
all
good to
of you listening in and everyone here on the ship. We definitely
driving
couldn't
out.
do it without our partners, especially our ROV team from the
Yeah,
Global
I'd like to move
Foundation of
30
Ocean
meters,
Exploration,
bearing 060 degrees.
and our
speed decimal 2.
videographers who create beautiful videos of everything we've seen and the images that you see on Facebook and our web
Good copy.
page and on Twitter and shared all over social media websites.
Move initiated. Thanks, Bridge.
They are quite talented.
Yep. And we have our data people, data managers that keep track of all this stuff. And the outreach folks that are making all these links and you know, these events and kind of public interest things possible. And we also have the entire ship's crew from the Okeanos Explorer, keeping everything running, getting us where we need to go, keeping the lights on, the AC on.
Looks like you have a little tether left, Pilot.
Copy
Might have
that.
to wait for the move to kick in here in a minute.
Copy. I'll try not to stretch out any further.
You have about 3 minutes left until you start moving.
Copy that.
So some interesting Beethoven facts right now are being shared on our chat room. Scott just discovered that in Beethoven's choral fantasy,
about a 20-degree
there's a
angle looking
line
at
that
you.
is flowers which eternally bloom. So we're thinking he was referring to these beautiful anthemastis corals. They look kind of like flowers, and they are eternally blooming here in the deep sea. Maybe he came up with that after he dunked his head in that cold water and sort of channeled our deep sea organisms right here.
Could be.
Could be. I think Beethoven secretly liked the deep sea.
Nav co-pilot.
Good, co-pilot.
What time did we put that ship move in at?
I put it in at 43.
Copy that.
So that would be 3.5 minutes ago.
Perfect. Thank you.
Yep.
I do believe that we're starting to move.
So we're seeing a lot of these
Copy
corbitellini
that.
sponges, these big leafy sponges that we were looking at earlier, all over the place. Still seeing lots of anthemastis spotted across the landscape here.
So I'd consider this a success for when we were targeting high-density deep-sea communities.
I definitely would think so, Casey.
So why is it important? For several of these days, we've been targeting high-density deep-sea coral communities. Why is it important to study those ones or to find them and locate them?
Well, we don't know very much about these coral communities, and knowing where to find high-density communities, we can more easily protect these areas and figure out what's here. There's an urchin, purple urchin we haven't seen before, right by the bottom middle screen. A lot of these deep sea organisms have been found to have compounds that they've been able to use for development of medicines and other applications. And if there is to be mining here in the deep sea, mining this manganese crust, we want to be able to protect these high-density communities.
Yeah, so by doing these studies of where we map in detail, get the topography and other bottom characteristics, and then actually go and dive or photograph or in some way sample the biology, then we can determine, we can kind of model or predict where these communities live, and then we can also predict where, like at the beginning of the dive, there wasn't such a dense community, and we saw that both yesterday and today, and then as we move up slope, we've found more of these communities. So we can kind of predict based on, the topography, the oceanographic parameters, where geographically these features are, what type they are, what shape, and we can kind of predict where these communities are, so we can really prospect for them. So we're getting an idea where they may be, and which will help with people that modelers, because we can't possibly visit every single seamount in the world. There's thousands and tens of thousands of them. So this gives us a way to, by looking at them in detail here, we can figure out where they might be.
All right, so Cirrus is starting to move. We can go pretty slow, Sean.
And we know that a lot of times these communities are relatively slow growing. So if there were to be a disturbance in this area, it might take a long time for this community to come back. And if too many are disturbed, it might be difficult for the community to recover, and that might truncate some of the genetic diversity across these communities in our oceans.
Right, and then I guess one field of interest is
Sean,
how
we got 100 meters
how
left.
similar or different
Maybe
they
90
are from
meters
one
left,
seamount
somewhere in
to
there.
another and one
Yeah,
part of
we
the
got
ocean
launch.
to
We got
another.
an hour.
Last pilot told me to book it.
Hour and 20. So.
Copy
We
that.
made
So do you
made really
typically
good
end up
time, so-
finding other organisms? So in these high density
time to
communities,
explore
do
all
they
these
attract
two
others that we
efficient
may not be
together,
seeing?
I think is the problem.
That's correct.
Well, that's a good question. There might be more stuff here than we do see. There's especially a lot of smaller things that we can't capture on our camera. In the sediment and under rocks, there are quite a lot of macro and micro invertebrates. There might be some fishes that live in this area that might be scared away by our ROV. The lights might be, they might be sensitive to that. And so they could be hiding from us. And of course, you know, what we do see are the sessile organisms generally because they cannot move from us. But those things that are more mobile, they are around here, and we might not be seeing them all at once. But that doesn't mean they're not here. Seeing a Pleurogorgia. I've been seeing some Pleurogorgia, which is a type of chrysogorgid. And there's just so much to see right here. That purple urchin that we zoomed in on just a little while ago, I believe that is a urchin in the family Echinotheridae. The species Tromicosoma hispidum. It is so beautiful.
That's pretty cool.
I know, isn't that neat?
Do you see shrimp? Place center on it and take a quick zoom.
Oh, we have a shrimp in view. Let's see what kind of shrimp he is. This little, oh, this has a very short rostrum, so that's that little projection between the eyes. So I'm thinking that it might actually be Bathysissimidae instead of an Aristiidae shrimp. So from the research that I've been doing about these deep water shrimps, there are two families that I've found of these shrimps that have these long pleopods, and those are those little arms that you see swimming out in the water. And the Bathysissimids have a very blunt, more robust and short rostrum, whereas the Aristiids have a thinner, longer rostrum. Other characters include seeing spines on the carapace, so that's that front part of the shrimp. So if you were to eat a shrimp, you generally use that part in making your shrimp stock, and people generally eat the shrimp tail.
So how would these shrimp taste, do you think?
I would assume they would taste like shrimp. Just like
Imagine
any more,
that.
yeah. Just imagine that shrimp would taste like shrimp.
They wouldn't taste like chicken?
They wouldn't taste like chicken unless you're in the family of people who believe that most things taste like chicken.
Okay.
Oh, this little
Well, look at that.
shrimp is gonna land.
I think he's
Maybe not.
He's showboating for us.
Now, to tell the difference between male and female shrimp, obviously from this angle we can't do it, but they have, the males have special first pleopods that are different. So on the underside you would see little, almost grasper, like
appendages.
Appendages.
Okay.
Yeah, and the females have a general opening type of thing. It's not. That's not a very good description of what it looks like.
I got the general idea.
Yeah.
Get Liz's back on.
Roger that.
I'm just trying to imagine like the diagrams. And some of those characters can also be useful in identifying shrimps.
Oh, look at this big sponge thing here.
Oh, wow.
Wow. I guess it's the same as the others, but a lot bigger.
Yeah, a lot fluffier.
Is there a desire for zooms at all, science?
These are the same as we've been seeing.
Just size? Is
I think it's mainly just,
just a very large one.
Maybe take a...
Yeah, kind of get a...
If we have room on the tether, maybe we can take a flyby here.
Yeah, an overview view.
Yeah, we can do a quick overview. Just gonna go slightly out of view. Copilot, real quick.
Copy that.
Just get a sense of
Let me know if you'd like me
scale
to
of
follow.
this community.
Copy.
Yeah, you guys are stationary, so.
Yeah, so this first one's probably like a meter.
So it's actually
wide.
two that are next to each other, rather than one long one.
Alright. Come back
Okay.
centered.
That's good.
What'd you say, Nav?
We're stationary. So.
Copy.
Thank you, man.
And we have time, so.
Okay. What will we see next?
Looking good, pilot. Got you back in my field of vision here.
Nice.
So far, this is...
Can we zoom in on this? It's hard to see, but next to the sponge, just to the right, there is a fluffy looking Chrysogorgid.
Like left.
Just barely to the left.
Just
It's
barely
not,
to the left.
yeah, it's between
Video
that patch of
partial?
Anthomastus.
The light pink thing?
Upper left, I think.
Yeah, it's the upper left.
Oh, okay. I see it.
Yeah, it was really hard to see. It sort of
So-
blends in with the background.
Yeah, there we go.
Up a little. Yeah, there we go. I think
Yeah,
that's
so
it.
this is, looks like a different species of Chrysogorgia. The polyps are a lot smaller than what we saw in some of the other ones. Very beautiful. And there are lots of little associates all over. Maybe shrimps or amphipods? Oh, they're amphipods. There is one that's in a really good view on the right branch near the top.
There.
Yeah, see, you can see his eye glowing.
Oh,
Yeah, the little,
Sorry, video lost it.
sneaky little guys.
yeah, so.
So what are they doing there, besides hanging out?
Well, they have shelter when they're on these branches, and being up near the coral polyps, they'll be closer to the food supply. They have something to hang on to. And there's a lot of amphipods here, so you know they might be in a group that might be interacting in some way. So to identify amphipods, there's a lot of characters that you can look at, but for the most part, from our view right here, it'll be hard to see. Most of the time you have to do a lot of dissections. but they are very cool creatures. They have compound eyes that you can see looking at you, almost glowing in our light from the ROV. Thank you, pilot.
All right, happy with that?
Yep.
Yep.
All right, video.
Maybe somebody who knows a little bit more about amphipods, they have more of a sort of bean-shaped eye than a round-shaped eye. I definitely think they are Gamma-Ridian amphipods.
Les is talking. This is kind of unusual. He was asking about collectioning. I just saw that.
Is there a desire for a collection?
There is mention of it. Let's see here.
What is he saying?
Stand by.
Might be related to Rhodaniridogorgia, but not sure. Looks like it could be a branched Chrysogorgia. Yeah, I guess we could grab it.
All right.
One branch of it.
We're in a good spot.
But I'm not sure if it's worth it right now.
Okay.
How far away from our final waypoint are we, Nav?
Yeah, watch leads. The ROV is 100 meters away, and we have an hour left, so we can very easily make it there.
Okay, thank you, Nav.
So I guess we could take the time to do this.
So to go for collection?
Go for collection. Let's hope some of these amphipods will stay with it.
Yeah, copy that.
Because I want to take a look at them. So it has sort of a battle butt brush look like Chrysogorgia geniculata, but it's branching in the middle, which is a little bit different.
This should hold. All right, co-pilot ready for hydraulics?
Hydraulics are spun up.
Nice.
Do you want me to take control of the mini Zeus?
Sure. Yeah, it'll be good.
Here, I'll get a look at the sample and.
All right. Arms coming live. 3, 2, 1. I'll probably stick this starboard.
Yeah, both starboard bio boxes should be open and vacant.
Copy that. Is that about what you'd like, wash shades?
Yeah, one branch of this should be fine.
All right. Alright, so co-pilot, next question is, can BioBox be opened here? Or do I need to move?
We can open up the drawer a little bit and just look. I don't think it's going to be able to open.
Yeah, I can move back.
Do you want me to try?
Oh no, the shrimp failed.
Yeah, I
Oh,
can
it's a
move
squat
real quick.
lobster.
Okay.
Video, you want to get quick views of this before we maneuver away? Yeah, I'll try to back up to a less populated area. Yeah, whenever you're good video, you can just come wide.
Ooh, we get to rock with it. No, we don't.
No, we don't get the
I
rock.
thought that from that angle. I was like, wait a minute. The
Sorry
rock
about
came up.
that.
Probably could have gotten that rock. It looks loose. Get
Well,
another
let's go back for
Anthemastus
it.
and a Chrysogorgid all in one.
Yeah, I'd go for that.
ROV nav, Bridge.
But this way that colony can recover and keep growing.
Go ahead, Bridge.
Yep.
Yes, I'd like to bring the bow 5 degrees to port.
Okay. Yep. Go ahead.
Okay, right. We're okay to do that now. So I'm coming.
So
5 degrees
this
to
evening
port.
when we prepare these, this colony might be a bit pokey, so we might need to put it in a different type of container.
Okay.
But we'll
Maybe
see.
you could put it in a poke bowl.
A poke
I
bowl.
was getting to that. I was getting to that. I just.
It's my favorite meal.
It's
Poke bowls.
That rock right by your port toe.
It's a type of Hawaiian food if you're not familiar, of ahi tuna or another fish. Sometimes they do it with crab or octopus on top of rice in some sort of sauce. It's delicious.
You could kind of turn to starboard a bit and put your side down.
Our wonderful
Those
chef
are
Ray
fish rats.
made some poke the other day
Not a
for
great
lunch.
timing for
It was
that.
great.
It's going to steal your coral.
Oh, there looks to be a fish. Checking out our coral sample.
Uh-oh. You better be worried, Les. You might.
Yeah, I
Go
think
for
it is a rat tail or grenadier in the family Macuridae.
Cobalt, you think that's good?
I think
we don't
can
have
try.
Bruce
I
on the
think
line
it's gonna
today,
hit on the
but
port toe.
from what he was
But,
telling
we can
about
try and extend a little bit.
us, telling us earlier about these fish is that I think it might be a
If not, I'll just kind of
Coryphaenoides.
lift and we'll do floating.
Coryphaenoides.
Coryphaenoides.
Is that an East Coast fish?
Oh wow!
This is
If
Bridge,
you
go
look at the view right there, he's just this fish is just right up in our bio box area.
That's correct. Bailout is west.
It's a little lonely.
The heading move is
Wants to be
complete.
friends with the ROV.
Okay, so that pushed us off a little bit. Pilots were
That is
coming
correct.
back in.
We are on station.
Alright, so I'm going to float, Copilot.
Copy.
Try to go into autos, and hopefully I can stick it in that.
Copy.
It's a beautiful
Keep your eye on
fish.
the- in your cam there, so I think we're okay.
Yep. Yeah, we've got good delta, good altitude.
Let me just consult my fish
Coming up with
animal guide. See if I can find some nice pictures that might help me be a little more confident about that ID.
Alright, yeah, go for
So
draw.
the Coryphaenoides tend to be a little
Copy.
bit thicker in the body. They're scavengers.
Watch.
They have a long a little barbel on their
Fully
chin.
extended. You like that view with the Mini Zeus pilot, or?
That's pretty good. Yeah.
Perhaps Coryphaenoides longiceros, or maybe there's another species. I just like the way that fish came and photobombed the collection.
Yeah, it took kind of, it was a little distracting.
Alright, go close drawer.
Okay, that's fully retracted.
Nice, thank you.
Co-pilot, that was starboard inner. That is correct?
Yes, starboard inner.
Alright, arms off.
Gonna center up the mini Zeus and then give you back control.
Nice, thank you. Autos helped great there. It's nice.
Okay, we're almost to the top of the cone.
Yeah.
Thanks, Mike.
Alright, Nap, what's ship status?
So we're holding station. We just finished heading change. We would be ready for a ship move whenever you want. The last one was bearing 060, 30 meters. How do you feel about another 30 meters?
Sounds great, yeah. We're stretched out nicely. I think we can move ahead.
Attention all hands. There will be operations being
Is that
held in
bridge?
the forward lounge.
David at the mic?
I repeat, operations being held in the forward lounge.
On sonar, Sirius showing maybe like slope towards, I'd say, like 0.
Hey, bridge, do you even have your mic?
30.
Roger.
Yeah,
Yes, we do. Sorry about that.
No problem. 30. Yeah, we can keep going 60.
60 is good?
I think that seems to be, yeah, pretty good.
That sponge right in the middle is really
so I'm gonna
cool
pull in
looking.
30 meters.
Yeah, I was looking at it. It looks like a sink.
Yeah, why don't you put that in?
060.
You know, I've always wanted one of these sponge. I feel like they would make really cool, like, furniture designs.
It's the bridge. Go ahead.
Like a bar stool, maybe.
Yeah.
That's going to be range 30 meters, bearing 060, speed 0 decimal.
I think the other day we saw a Sacco Calyx sponge. I feel like those would make really neat lamps.
Coming up.
Video, do you see fish?
Oh, there's a very small fish.
Video, go for zoom
Possibly a different species of rat tail or grenadier.
Would that be a juvenile or just a small fish?
I'm not sure. It could be a juvenile. It's
Video,
probably
go
a
for zoom.
different species
fish.
and genus than the one that just photobombed us.
ROV nav.
Yes, this looks
move
like
initiated.
Kumba.
Copy, thank you.
So it is a rat tail, a fish in the family Macuridae, and the genus Kumba. I like that name.
Kumba.
Kumba.
So for folks following along on shore, if you're looking at video feed 3, we've updated the position of D2, and as you can see, we're on this point right here is D2, and we're just about to the summit of this cone where we've encountered some high density coral areas. Just wanted to give everybody a kind of perspective of where we are. Again, we're diving on Beethoven Ridge, which is this larger feature you see. We're on top of this cone feature on top of the ridge, and we're just about to the top.
Alright, video, I think that's pretty good.
So,
ready, you can go for,
so stay tuned. We will bring you the summit.
Once we lose this fish, can we look at some of these corals? There is a black coral right on the rock in front of us by the lasers.
Yeah, is it pretty much dead center right now?
Dead center.
Yeah, video, come back in. The little one right here?
Yeah, that little one. I think this is Umbellapathes. Now Tina's gone to bed.
And a crab.
I am fairly certain that's what this is. Beautiful.
Copy that. Yeah, that's kind of what.
Les said he spotted a bamboo coral.
Settle real quick.
That's good on this coral.
All right.
Can we see the white coral that was a little further down?
White coral further down.
It might be a bamboo coral. Yeah, this one.
Right here. Nice. All right, video, go for zoom.
I think they're actually primnoids. No, that's a primnoid. Thank you, pilot.
All right. All right, video, come wide. Yeah.
This sponge is particularly photogenic if you want to get a nice
did the shift change?
overview of this area.
Oh.
You're stinking though.
Yeah, I didn't realize it already happened though.
It's like a giant vase.
Say when, pilot.
Yeah, science. 1 second, we're going to do a quick pilot change.
You're in good shape for now. Keep an eye on you.
Nice. Copy that, man.
And if you're on shore, if you change to video feed number 2, you can see a nice overview of this landscape from the Sirius, looking down at our Deep Discoverer ROV and how it's checking out this very large coral, sponge right now.
Sponge has to be pretty old.
So if you're just tuning in, this is Megan Putz, your biology lead.
And John Smith, your geologist.
We're coming to you live from the Okeanos Explorer up in the Musician Seamounts. Today we are exploring the top of Beethoven Ridge, and we are headed to the top of a small cone feature located on this ridge. And so far throughout our dive, we've been seeing high density community of Anthomastus corals. Those are those little orange corals that you see spotted all over this landscape, and sponges. That's that giant sponge right in the middle of our screen right now, along with some primnoid corals and many others.
Yeah, and these seamounts and ridge features have really not been explored in the past. There's been some mapping done, and there's been some rock dredging done for geochemistry and age dating in the past, but to our knowledge there's not been any observations like this with manned or robotic submersibles or deep sea cameras to actually capture what the bottom looks like and explore the communities of biologic activity
pilot.
And
Got about a 40
different
degree
organisms
angle
that
down
live
on
here.
you.
So this is, you're seeing this for the first time
Are you
that
feeling
anyone's
any
ever seen
current?
these things.
That's right, John. We've never explored down here, and this is the first time anyone has ever seen this place. We are currently at 2,320 meters in depth. How far is that in miles, John?
That's like over 1.5 miles, getting close to 2 miles deep.
That's pretty deep.
Yeah, you put some pressure on me there with that calculation.
I certainly did. Jokes. And at these depths, the water temperature is really, really low, almost near freezing, about 2 degrees Celsius. But that cold doesn't stop these corals and sponges from growing.
Or the shrimp, or the fish,
shrimps.
or the urchins.
They actually really like it.
Yeah, the problem is when we bring them up, they don't like the warmer water and the sudden lower pressure. So that's a bit of a problem. On this vehicle, they have a nice setup to keep the water that we collect cold until we can get it up to the surface and on board. But being in the tropical ocean, and especially in the summertime, the surface waters are in the low 80 degree mark, so 80 degree Fahrenheit, and if you're bringing it up from just over 32 degrees Fahrenheit, that's quite a change. It's a dramatic change for anybody. Imagine if you suddenly went through such a change.
Video, can we snap?
It's like if you're wearing your full, snow suit in multiple layers, and all of a sudden you ended up in Hawaii.
Or in the desert somewhere.
Video pilot. They're right on the edge of a Some of the trailers.
So a lot of the organisms, I believe, that live in water, they're more susceptible to smaller temperature range changes than those of us who live in air.
That's right. If you've ever been swimming in a pool,
didn't
you
you
notice
go
those
around
small
the big one?
changes.
What?
I did a little bit.
In a hot tub, you have temperatures of about 100 degrees, but then once you get into water about 80 degrees, you're thinking, oh, this is actually kind of cold.
Hey, Bob, can we
Right.
see?
So that's why when we talk about ocean warming, global warming, and it's in small changes in the ocean relative to what we experience, it's kind of a big deal for corals, especially shallow corals.
That's right. Right now we're zooming in on a chrysogorgid coral called Pleurogorgia.
It's pleura because it's got a lot of gorges.
Or
it's really gorgeous?
It's really gorgeous.
Because it's pleurogorgeous. I have to use that
It's
line sometime.
pleurogorgeous.
I don't think she'll get it though. You're pleurogorgeous.
You're pleurogorgeous. Well, you have to talk to a deep sea coral biologist.
I'm sure that line gets used a lot.
I don't think
will now.
It will now. The world has heard it, and thus it is a thing. These Anthemesses seem to like this broken pillow down in the right-hand corner of our screen. Those are the mushroom corals. Those orange little tree-like, flower-like corals that you're seeing on that rock. We collected 2 of these today. They happen to be on a cobble that was just small enough to fit inside our bio bin.
Yes, we wanted to preserve the rock as best we could.
Indeed. That rock needs to be protected and cared for quite a lot. But actually, these Anthomastus corals are soft corals, so they're relatively fragile, and we want to make sure they were protected on their way up to the surface. Oh, there's a, looks like it might be an anemone. Can we zoom on the anemone that is just to the left a little and down to the lasers?
Yeah, video, we can frame this one up as we approach.
And there's a mushroom.
We can take more zoom video.
Looks like one we saw yesterday.
Yeah, it's really interesting looking anemone with its mouth closed. That's that projection right in the center. And anemones are hexacorals as well, just like black corals.
Ah, yes, someone on the chat pointed that out earlier today. Did not know that.
Co-pilot, you should have already stopped swinging or be pretty close to stopping.
Copy.
All right, could we pan left?
I think we're starting to settle out.
Yeah, it looks like there's a closed Anthomastus.
Yeah, that's what I wanted to show, so we could show people what those look like, right in the center.
Yeah.
The little mushroom nub, right?
Yeah, they can pull their polyps all the way into the main stalk of the body, and it looks a little bit like a mushroom.
Yeah, so these are the same thing. That's what
about
retracted with
looking down on you, pilot,
which
but
is right above, fully extended. Same what we've been seeing this whole trip up the cone.
Okay, video when you're clear, you can come out.
So when we get them up on our onto the surface, those polyps are probably going to be retracted. There is a really interesting coral colony on the left side that looks a little bit convoluted.
Please
and it's a
pardon the
fan.
interruption. Your conference contains less than 3 participants at this time.
It's right in the center.
If you would like to continue, press star 1 now, or the conference will be terminated.
Okay, video, you can frame it up as I approach. Did you need a closer look at this one, or would it snap zoom?
Let's snap zoom. I think it's a hemicorallium, or a pink coral. We saw one of these a little bit earlier. Yep, hemicorallium. Very beautiful. They actually use these corals in jewelry making. So if you get pink coral jewelry, this is the coral that those pieces of jewelry come from. In Hawaii, there's actually a fishery for corallium coral. It's not currently active, but it is a managed fishery in Hawaii.
Or the conference will be terminated.
So how old would something like this be?
This one is a little bit smaller. If we can zoom out, we can see the lasers. The lasers are 10 centimeters apart, about 4 inches. So this looks to be about, what do you say, 6 to 7 inches. Something like this
sea cucumber right to the
might
left.
be, I want to say 40, 50 years old.
Wow.
Let's look at that sea cucumber. It's a little translucent on the rocks. I think this is our first cucumber of the day.
The conference contains less than 3 participants at this time. If you would like to continue, press star 1 now, or the conference will be terminated.
So these deep-sea sea cucumbers are often called sea pigs, and they have these little tube-like feet that they use to march along the bottom. I really like the term sea pig. Do you know how to tell the difference between a land pig and a sea pig?
No, I don't.
A land pig, bacon from a land pig tastes delicious. I don't know if we can get some bacon from this sea pig.
Conference is being terminated.
Pilot, as Nav had
So-
mentioned, I think we've settled out.
What about
We're in
it?
a pretty stable position here.
Copy that. Thank you.
No problem.
So this sea cucumber is in the
If you have any more
family
Zoom, you can take it.
Elliptidae.
You're not busy with the conference call.
Oh, that's a nice view. And it has a little, what do you call, sort of a crest around
Oh.
its neck area that has
Right.
projections on it. And our sea cucumber biologists will use that character to be able to identify it further. Though I'm not sure what this one is any further than the family Elliptidae.
Okay, video when you're clear, you can come out.
Common name sea pig.
Sea pig.
The sea pig.
So in this case, would you say when pigs swim,
When pigs swim,
you'll believe it?
I'll believe it. Now these sea cucumbers have been known to swim. They're not pelagic, so they don't swim all times, but they can swim.
All right, so it's more believable. Believe a sea pig rather than a land pig.
The graceful movements of the sea pig.
Hey, pilot, so that's me. I think we're stopped. No more swing, but I'm looking at you. 8-5.
10-4, making forward way. How much time we have left on bottom for now?
We have about 40 minutes. Oh, there's a nice one right there, far away, top left.
Large white colony.
Yep.
Yeah, we'll get there. You want that pirouette, don't you?
I do.
I'll see what I can do for you.
All right, sounds good.
There's so many colonies here.
Yeah.
Trying to find something new we haven't seen today. I feel like a Where's Waldo.
All right, sorry about that, folks. We were having a little trouble with our conference call, but it seems like
What about that
we
tiny
are
little
good to
black
go.
one that's right in the middle of the street above the lasers?
That's what I was looking at too. Possible spot right away.
Seeing a nice Paraclyptrophora
Nice white.
on the bottom left-hand side of the screen. A lot of these Corbitellinae, new genus, sponges.
I'm not here.
You're not hearing me? I guess we're not connected. Oh, can we look at this, pilot?
Yeah. So I think we're
This
looking at
is
like a
crinoid
dark
on
stout
a-
crinoid possible. Video, can we zoom in this?
Yeah.
We're kind of looking right at it. Oh, he's hanging on to it. That's what's going on. Okay.
Ah, yes, very cool. We were calling this Cerametra for a
Wow.
little while, but it has a real name, not well, a different name than that. Cerametra is known from the Atlantic, and this one starts with a P. when I can't remember how to pronounce it. So I'm going to have to read it.
I might try to spin around. So
Looks like
Sathrometra. Sathriometra.
Wow. Okay.
I feel like Cerametra was easier to pronounce, but this is Sathriometra.
Okay, video, you can zoom out full wide. Keep looking here. Thanks.
Looks like one of those evil spaceships from science fiction shows.
And that was on, looked like the skeleton of a Bathypathes. And that's a black coral, so you can see that skeleton was really black. On the Bathypathes that we collected yesterday, when it was alive, the tissue was a bright yellowish, orangish brown color. But without that tissue, the skeleton is black. And since earlier I was talking about corallium and how there's a fishery for corallia or pink corals in Hawaii, there's also a fishery for black corals, and that's that Hawaiian black coral that you see at Maui divers. So when you go to Hawaii, they sell black coral jewelry. And that jewelry is made from black corals that specifically grow in Hawaii, known as Antipathes grigii or Antipathes grandis. And these black corals get very large and have extremely thick bases, which allow for the making those nice pieces of jewelry. And the fishery for black corals is managed so that it's sustainable.
In Hawaii at least, right?
In Hawaii at least. Around the world, I cannot say the same.
Well, it looks like we're probably at the top of this cone now. It's flattened out. Look how
Oh yeah, we're almost
the
to the top.
topography is.
And even at the top, we've got some massive sponges. These sponges are quite large. So we'll get these lasers on our sponge and see how big it is. Those lasers are about 4 inches across, 10 centimeters. So that looks to be about a meter, I would say.
Probably so, yeah.
And this is a 3-dimensional sponge, so that's quite a large sponge.
I think we have found our colony.
Yep.
Pilot. Perfect colony for a pyramid.
In the background, I see some Paragorgia. There are a bunch of primnoid corals, possibly in the genus Narella. Lots of Anthomastus just chilling out on almost every rock you can see. Our Corbitellinae sponges. That's this big sponge and all these little sponges in this area. I am really excited we found such a dense community today.
Yeah, it was not quite like this yesterday. We found some more denser corals, not much sponges yesterday, but nothing like this. So I think first day we were out, first dive was pretty good. This one may be better. I don't know. Some of the corals don't seem to be quite as big as I remember from the first dive.
Yeah,
the sponges
that
are
first
definitely
dive we
bigger.
saw a lot of bamboo coral.
Yeah.
But this dive, we've only seen a couple bamboo corals, and they were that big.
Yeah, okay.
We've seen a lot more of these primnoid corals, though.
Okeanos is back.
And every community is a little bit different. But there are also a lot of similarities between them. There's something tall in the background.
We're probably getting ready for a move. Yes, think. Yeah, so
It's
let's
absolutely
see.
gorgeous.
Looking at you, 5-0. It's in a nice standard position. Fernando, ready for a move?
Okay. All right.
Thank
Thanks.
you.
So, yeah.
So the conference
Straight
line is
ahead.
back
I
up
think
and
this-
running. If you would like to
The last
call
one
in
was
any scientists on shore.
060.
Yeah.
30 meters.
Still looks good to me.
Sounds good.
Co-pilot is a go.
All right.
For move.
Pilot is go.
Copy that.
Go ahead, Nav.
Let's get the single range. That's going to be range 30 meters, bearing 060, speed 0 decimal.
Copy that. 30 meters, bearing 060, speed decimal 2 knots.
Good, copy range.
Move initiated.
Copy.
So my dad just emailed me, and he was wondering what our dive track is like, and what the predominant current flow is, in what direction it is in this area. So right now, our dive plan started at the base of a cone on the western side of this cone feature, and we headed east up the top of the slope to the very top of this cone feature, and we are currently probably just a few meters from the very top.
Yeah, I think I overheard them at 30 meters to go.
Yeah, 30 meters to the top. And the current flow today has been relatively light. At the beginning of the dive, it was flowing from the so north to south, or south to north. It's from the south to the north. We are currently at a depth of 2,311 meters.
Drift slightly starboard.
On Beethoven
Out of my
Ridge.
view. Can rotate or just kind of hang out there too.
So we'll just see that one large column here, and then it's going to come back to center line.
Copy that.
We probably do need to collect a sponge for Chris. Are we allowed to collect 4 biologics? Or can we count those Anthomastus as associates to a geologic specimen?
Thinking of going for another rock.
Well, if we can get one with one of these Corbitellinae, new genus, those little vase-like sponges on it, that would be great.
All right. Do you see any?
I see tons of Corbitellinae.
Okay. With a rock, though.
So these, yeah. I don't know which ones are on loose rocks, though.
Okay. I don't see.
I'll keep my eye out.
Roger that. Yeah, I'd like to get
the sponge we're looking for is the one. I'll get lasers on one to verify.
Yeah.
It's kind of.
Yeah, it's that kind, like the vase with little weird projections.
Copy. Keep our eyes open. Nav, which box is open? Starboard.
Should be starboard outer.
So it's a little bit larger bio box.
Did we put anything in the port rock box?
I don't believe so. We only have one official rock sample in the starboard, right?
Yeah, I think port
So
rock box is
I think
open.
a sponge like this, as long as the rock doesn't roll around, would be okay.
Okay.
If it came up in the rock box.
Tether looks good.
Otherwise, the other open is the starboard outboard, or
Maybe.
or is it the port?
Yeah, starboard outer.
Starboard outboard.
Yeah, if it fits in that, it would be better. But
Yeah, it would be better to keep it in the water.
so if we find one of appropriate size.
So that sponge in the back could be two things. Perhaps a polyopogon that looks a little odd, or maybe one of the rosellid sponges in the family. Trying to pronounce it with the L. Langunelidae. Yeah, I think that's good enough. Languinality.
Oh, sounds good.
Oh yeah, it looks like a vase, so it's probably that one. But who knows? It could be something different.
Something completely different.
Something completely different. We need our sponge expert on the line to be able to tell us a little bit more about interesting sponges, but this one definitely looks interesting to me. It's very, very beautiful.
Looks like elephant ears.
Almost.
Or Mickey Mouse ears.
I like the way the opening of the vase is sort of curling under. And just like the texture. Could we zoom in on the texture?
Yeah.
Side of this vase? Oh, and there's a little tubularid hydrozoan on this sponge.
Can you zoom in video?
That is really cool. Well, now that we've gotten a closer look at it, it doesn't look like the Langunelidae.
All right, pilot.
There's some discussion if we should collect some of this. I think these big sponges.
Nice steady shot, pilot.
Yeah.
You don't.
I don't know.
Can zoom out video, frame it up maybe.
So for sponges, we just need a small piece to be able to tell what it is, because every sponge has a very unique set of spicules, and those are the glass pieces within the sponge skeleton that make up that structure of the skeleton.
Move complete.
Lasers are off.
I almost don't want to destroy it. It's so beautiful.
Yeah, it's very unique.
Nice.
Let's see. What other things could this be? Oh, it might be this Hexactinellida
Oh, actually,
vase.
this is enough. Just
What
want
do
to
you
let you
think?
know
Does
that
it
we
look
have
like
about
that?
26 minutes of the dive.
So I'm looking at my animal guide, and it looks like what we have identified as just Hexactinellida in our animal guide, which means we don't have a very specific identification for this particular sponge. Our reference picture has that sort of convoluted texture on the outside of the vase. And even this sort of lip is forming on our reference photo that we can see fully in this full colony. I'm loving this 360 view we're getting of this sponge colony.
Yeah, it's quite impressive.
Watch your
watch your port.
How do we sample this if we did want to? Do they just pull a piece off, or?
Yeah, they would need to pull a piece off. The only thing is sometimes with these sponges, the spicules can be different from the top and the middle of the sponge, so we would need to get a large enough piece to be able to make sure we had all the different types of spicules. But here's a up-close view of the lip of this sponge.
Zoom in on
It looks very like the one that we're looking at right now on our screen.
All right. Yes, no, maybe.
I want to say yes. I don't think this has been sampled before. It was last seen in the Marianas.
Okay, you can come out full wide video.
Does anybody on the line or on our chat room know if we sampled something like this before?
I think from what I've read, nobody's really sure, but it seemed like there was a interest. Yeah, nobody really knows for sure. There's some ideas floating around.
Yeah. Well, I don't want to not sample it and not see it again.
All right. I think we have time.
Okay.
We'll get a piece of this.
Let's get
pilots?
a nice piece of this. So we don't need the whole sponge, but we want to get a decent chunk from the top and sort of lower edge of the middle, I guess.
That would be starboard outer.
Top.
So
where the lip is folded over or somewhere else?
Yeah, I'm not sure what would be the best location.
I'm worried that if I try to grab it, I won't be able to break it off. You know.
Yeah, I'm not sure if this is a soft sponge and it'll bend or if it's crunchy.
Yeah, we might risk topping
I'm
the
leaning
entire
towards
thing.
it might be actually relatively stiff.
Yeah. I could.
So there's some concern about knocking it over completely if there's.
If I go in for the grab, I'd knock the whole colony over, or I could try to snip some of this. Try to get lasers on where I think I could snip.
Yeah, I think the best thing would.
Zone up here, way up in the front. Because it's thinner, I think I would have an easier time breaking part of it off without toppling it. But I have a suspicion it's pretty stiff, in which case I would have a pretty difficult time. Yeah. I can attempt it, but there is a risk of...
Yeah, well, this is too big to fit on our box, so we can't take the whole thing. Let's just get the best piece we can, and I'm going to leave it up to you, pilot.
Okay.
With the minimal chance of knocking it over.
Yeah.
So we'll compromise on maybe what we would really want to preserve it.
Hydraulic spun up. And we're going for starboard outer. Drore's ready to extend. And nav ship stopped, right? I might still swing a little bit, but I think we're okay.
Yeah, that is correct. Ship stopped. You still might be swinging.
Copy
And
that.
the great thing about collecting something like this is we are going to get genetics from it. We are partnering with the Ocean Genomic Legacy, or OGL.
What camera view would you like up?
Mini Zeus.
And we'll be sending our little sub-samples to them of the genetic, or some pieces from the sponge to them. And
They're at Northeastern University?
yeah, I think so.
Yeah, hydraulics up, ready to go. Arms live.
And that'll add to our knowledge of all of these deep sea organisms that we've been seeing.
Do the sponges
regrow? What you said is your best bet. Try to
I
snip the top.
do believe they will regrow. It's going to take a while.
Claws
These
get it.
types of
If
organisms
you don't
do
cut it,
grow
you just kind of
relatively
grab it.
slowly, just like the
I mean,
sponges,
depending on the strength
just like
of
the
it,
corals.
it might just kind of tear it down. So I think to try to do a small snip is the safest.
And just touching this and breaking a piece off will give Chris more of an idea of what kind of sponge this is, whether it's soft or if it's more of a hard sponge.
Yeah, you know what you're doing. That's good.
Because we can try to use some of the
You can't tell those types of things just by looking at it.
So you're saying it could be a squishy sponge or a crusty sponge?
Crusty sponge.
Crusty sponge.
Crusty sponge. We don't know which team it's on right now.
Is this a good location to sample from, Watchley, do you think?
Yeah, I think so. That looks good.
Okay.
I think you have something, just kind of...
I think you cut it.
Took a chunk out.
Try to...
You have some in your jaw, though.
So I think we might have a little tiny bit in our jaw.
Looks like, there's a little bit in there. But
Yeah, we might need a little bit more.
Yeah, I think I kind of broke a section loose there. So I could drop what I have to attempt to grab a little bit larger piece.
Okay.
If that's what you would like to do. Yeah.
Unless that's big enough.
It's actually almost stuck in there, so you might be able to keep it.
Okay.
Yeah, just.
Well, it looks soft to me.
Move Jesus a little bit.
Yeah, you want it up.
Yeah.
Up and over. How's that?
That's good. Try to get that larger piece. Come out from this way.
Yeah, I think it might work well.
Oh.
Oh no.
Definitely got more of it though.
So is what I have in my hands enough, or I could go back to try to grab a larger piece? The options here.
I want to say we need a little bit larger piece.
Okay.
We can try to grab from the same area.
Starboard.
Yeah, drawer.
And then I think we can open the drawer here, so I could actually drop this part in and then go back for more.
Yeah, I'm extending drawer
Okay.
now.
That's good. I think that's it's gonna work. That way we get something, but we could get even more. So.
Yeah. Drawer looks opening nicely.
How big would you say this
slightly.
sponge is? Like
A foot or so across, and then several feet high, 2 to 3 feet high.
Larger section.
I like that.
Yeah.
2.5.
I think D2's stable again though, but it kind of moved slightly, maybe a foot.
You think the drawer?
It's tough to say. You didn't come back, you're moving laterally to starboard. Looks good.
okay, that looks good.
Follow me on Minizus.
Yeah.
As long as it stays. Oh no.
Stand by to catch up. Careful,
Oh,
you might
float
be
into the
close to
box.
this,
Do it.
camera.
Oh.
I tried to kind of get it close to the vehicle.
It was close.
I know.
Here, I'll come back.
Kind of reminds me of the 2001 Space Odyssey when the
careful
astronaut
next
floats
time
away.
coming back how high you are.
No.
Because it is possible to hit.
Reaching with the claw.
Tighten them and use this.
Pick them up.
Okay. I'm going to use grip lock on this next grab.
All right.
Sorry, but this is being real funny.
That's right.
Alright, there you go.
Perfect.
All right. Looks like we got it.
Gonna hold my breath.
You have the sample.
Hour.
Going.
And then, yeah, use your nip and then this one. You should be good. Yes, you have to like come in and then.
Oh, I think we lost it.
Yeah, part of it was lost, but part of it's still in the fingers, I believe.
Yeah,
Okay.
there's still some in there.
Coral cutters cut that section. We do have a small piece, I believe.
Yeah, it's in the box.
Watchley, did you see how much fell in the box?
Yeah, there's that's a decent sized piece. About a hand size, you think? Or is it smaller?
Maybe a little smaller.
Maybe a tiny. The
was-
other piece landed on the rock box ledge next to the little scoop.
That might be tricky.
I'm not sure if that's recoverable.
Watch these. Would you like another grab from the main sample?
Yeah, is the size of the box sufficient, or I could go for a larger piece if you'd like?
I think let's grab just one more piece.
Yeah.
I want to make sure that there's enough for the Smithsonian and the Bishop Museum.
All right, coming back.
If there's 2 pieces, then 1 gets each.
Yeah, close. Yeah.
It's gonna be 2 small pieces. Is what we got.
That'll be fine.
Okay.
And then.
And it looks like we probably got something more from the middle, which might have a different spicule combination than the area over by the edge, which is important for identifying this particular sponge. And we've gotten some really great imagery of this sponge too.
That's too bad. The larger piece did blow away, but I do have a small piece in the jaws again.
It's kind of stuck. Yeah, it's loose. Ooh, it's sinking. Nice, that was tricky. You want to close that?
Yeah. Sorry we couldn't get you a larger piece, Washley.
That's okay.
Seems like when I close the jaws, it cuts it in every direction, and then all that's left is what's in the jaw.
I think that's sufficient. Thank you.
Okay.
All right, both retracted. Arms off. Valve packs disabled. Maybe down HPU. HPU disabled.
That was a tough sample.
That was tough. Good job.
Yeah, you did good. There we go. Yeah, Cirrus didn't really drift much. Just kind of back where you were. I am looking straight down at you though.
Copy that.
Watch leads, just to let you know, we have about 10 minutes for the dive to be over.
And you didn't knock the sponge over.
Thank you,
so
Nav.
that's the
Okay.
most important part.
We'd still like to collect one more rock, and whether or not it has a sponge on it.
It's probably going to have something on it.
Yeah, so we
Just
can.
looking at this area, every rock has something on it.
So maybe I'll let Megan pick the rock.
Oh, I'm going to be biased towards sponges.
Well, I get final approval.
Okay.
And we'll even count this as a rock sample.
Okay.
Hey,
There's a bolosoma sponge on that rock over there. That's a pretty neat sponge. But we do know what that is, so we don't need a specimen of that one. I'm going to be biased towards these little Corbitellinae that we've been seeing all over the place.
How much bottom time we have, Fernando?
9 minutes.
9 minutes. Okay.
I don't know if we'll
we're gonna have to choose a rock pretty quickly.
really quick.
Okay.
In this next, in my field of view here, in order to have enough time to stow it. And,
how about center screen in that sort of jumble right above the lasers?
Yeah, I'll get closer to it. Let's see what we got.
I'm not sure if any of those are loose. Everything here looks a little bit loose to me.
Yeah, I think we should be able to grab most any of these. Just like that. I don't know. No, we already have that.
Is that rock too large?
It's doable. I'll give it a poke. It might be
This
two
one?
rocks.
The one the lasers is on?
Yeah.
Oh,
That might
okay.
be. It looks kind of big. About like
Yeah. No. So be it.
that looks like the ultimate sample right there.
Yeah.
That would be port forward.
The bonus.
I think Nolan said he'd carry that.
Yeah, he did promise.
It's a gooey rock.
Mhm.
It's going to be a gooey rock.
Gooey rocks.
Gonna have to wear a lab coat or an apron.
Probably both.
Right, hydraulics are spun out.
Or a garbage bag.
These are loose.
Yeah, do you want
You have to go port.
pour forward? Yeah, so you have to open the lid.
You want to wing it a little bit?
Yeah, absolutely.
So for those of you
wing
shore
coming
side, it'll be
inboard.
about 10 minutes. We're going to grab a rock. We're going for this one
She's in.
full of stuff right in front of us. We're going to put it in our covered rock box. So hopefully our collections will stay relatively healthy on their ascent.
Nice. Do you want me to ease this up?
Yeah.
Yes, it moves.
Perfect.
I
I picked a good one.
All right. Oops.
Vehicle's a little more stable here.
Might be settled out.
I think.
Yeah. Okay. I don't know if you have the reach. You might still have the reach. Kind of push it away a little bit.
So this would be
There you go.
06, I think.
Yep, 06. We did ourselves today. We've collected every single specimen we were allowed. And Tina will be excited to know that we got a bonus Anthomastus.
Just out of my reach.
Yeah, now it's definitely out of your reach. That's right, you can do a quick adjustment. There you go.
How much time I got, Fernando?
Plenty of time.
I'll come down a mini Zeus. Yeah, should be able to grab.
Let's go straight into the box, I think.
Yeah.
It's a large one.
Turn it a little bit like this so it doesn't hit the... There you go. And a little bit backwards maybe. Right there. Just go all the way down.
Mini Zeus come down.
There you go, perfect.
Just be careful with jaw. There
We
you
definitely
go.
got a big enough piece of that sponge for
All right.
identification. Great
Port wing
job.
coming
Great job.
out.
Record recovery.
Okay, time to go up.
Hope those guys hold on for the ride. Surface.
That was
Yeah,
port forward, right?
Yeah, port forward, drop high.
Port.
Yep, the port rock box.
Yeah.
Actually, I need these on 4 lights.
With that, we're leaving the bottom.
All right, so that is the end of our dive today, folks. We will be leaving the bottom. We had an amazing dive today, I would say. A very dense community of corals and sponges. Some interesting geology as well. Transecting up the slope of this cone feature on the top of Beethoven Ridge, up in the Musician Seamounts.
Yes, it started out kind of quiet, but it was a real choral crescendo and sponge as we rose.
High
along.
five for your music terms. We're going to become musicians by the end of this cruise, learning all about music theory and musical notation.
I don't know about that.
Well, I'm
We'll
going to
try.
Do you play an instrument, John?
Yes, has been a while. Percussion.
Oh, me too.
Yeah?
Yeah. I do mallet percussion.
Mallet percussion?
Yeah.
Lights are
So, like
See the
xylophone, vibraphone,
Ah, okay.
marimba.
Right. Yeah.
I also played bass drum in the marching band.
Oh, really?
Hmm.
Yeah,
I played snare drum.
So I'll come directly underneath you if you want to go to 090 and get a 20.
It's hard to
delta,
march
and
with
I'll
a marimba.
You will probably
And choose a xylophone.
stream northwest as you're coming up.
Okay.
Well, thank you for joining us, folks at home.
so
I
that
hope
puts
you
me
enjoyed the
in line
dive
with the
today.
ship.
Yep.
Just like that.
And thank you to our wonderful ROV team for the great piloting
Delta
and
20.
collections.
Right. Very much so, and everybody else
there.
involved,
Nice.
and to the shoreside scientists' help we had, and we'll be back here again tomorrow morning.
And every day after that for the next 20 days.
Push forward.
Let's get excited about this.
All right.
Tighten your-
Yep. So we'll be back with that.
pretty much directly underneath you, I think.
Yep, heavy and tighten. Looks clear ahead. Look at that.
Good.
Tether looks good.
Copy.
I lost you in main HD. Still maintaining Delta 23.
I'm going to remove them out of depth. Pushing out.
Copy that. Looks like you're pretty stretched out.
Pushing slightly.
Yeah, we're good.
Up and out.
Alright. I will remove auto heading.
Sounds good. Tether looks straight in my tail cam.
Yep.